to keep the camera
If we think perpendicular will give even better.
Hey everybody, good morning.
It's gonna give you the same.
Welcome back to the dive stream. You're watching dive 741 for ROV Sebastian. And you're watching a live high definition feed of the science camera on ROV Sebastian. And we have just touched down this morning again at Biobio Canyon. Currently it's kind of hard to believe at this specific location, but there is a storm brewing outside this bay, so we are completing some contingency plans for sampling, and we have decided to characterize this canyon. And so we are continuing to do that at this site this morning. Just like yesterday, the visibility is quite low, but we're not going to let that stop us from taking push cores and other types of samples. And right now piloting, we've got Tyler and Alex. And they were just deciding which way to go because to optimize visibility basically because of the current. So that's probably some of the chatter you hear going on in the background. But luckily here in our protected area, the waves are not so bad at all. And yeah, I see we have some viewers here on YouTube, Facebook, and Twitch. If you're watching, feel free to ask us any questions or let us know where you're tuning in from. And we're hopefully going to be on standby to answer all of your burning questions about the sampling that we're doing today. And with me here in the control room, I've got scientists from all walks of life and from many different institutions around the world. And with me here in the watch lead seat this morning, I've got Dr. Lisa Levin, who will be helping narrate this portion of this morning's dive. Were those the lowers? Great. Over to you, Dr. Lisa Levin.
Hello, everybody. Morning, afternoon, evening. We have just arrived at the bottom of this Biobio Canyon. We're at 699 meters water depth off the city of Concepcion in central Chile. We're fairly close to shore because we are trying to get out of the really bad weather that's out there. So we're behind the lee of an island near the head of the canyon. And as you can see, visibility in canyons isn't usually great near the head of the canyon. So there's a lot of suspended particles here. We are about to start a video transect. Whenever you say start, I'll have the loggers record.
Alright, almost there. Even if you down 45. I think, yeah. Because I think if you go slow, it'll hold it okay. Smooth it out. That would be better. Because that thing's, we're in another.
And
are we really?
these video transects are for our canyon specialist colleagues, some of who are on leg 2 of this expedition. And they've asked that the loggers record the organisms they see along the transect. So we'll try to be calling out things as we see them. Everybody. I'm not too sure how successful this will be given the really poor visibility, but we'll give it a try. We didn't do any video transects yesterday, but I think we're going to try today. And then Patchy at the end of this transect, if you like the coring spot, we'll go ahead and take push cores. So this particular expedition was a combination of 3 proposals that came into Schmidt. So we have a really diverse group of people. And one of those proposals was focused on canyons, my understanding. And so although we are studying seeps on this expedition, many of the people are, we also have several canyons in our program. The Chileans are eager to get a first look at what lives in the deep parts of their many canyons offshore, and to have them mapped. And we've been mapping overnight. So you're seeing the products of that map, I think, on one of the parts of the display. And
All right, start the line.
okay, start transect 1, please. We're at 699 meters. And we can see a bottom full of burrows. Those are the holes, some small and some fairly large. Those tell us that there's a lot of animals living underneath the sediment. And I'm guessing, based on other canyon cores, some could be burrowing sea cucumbers, some could be irregular sea urchins. There can be subpunculin and echeurin worms, probably large polychaetes, maldanids. It would be fun to have some sediment cores from here. See what's under there. There might also be burrowing shrimp, although shrimp tend to make sediment mounds, little volcanoes on the surface, and we're not seeing a lot of those. So you can see there aren't a whole lot of animals living on the surface that are visible, and that probably tells us that conditions aren't great for surface dwelling species. Mostly we have deposit feeders underground. These are 10-meter grids, right? So we have to go 10 of these. Yeah. But it's always interesting to have one look at the canyon axis before we start up, which most of this dive will be climbing the south wall of this canyon, the Biobío Canyon.
Good luck.
Something just floated by.
Where you find your way over 5 meters, like right across. This big wall sometimes.
I see some animal traces, like something's been crawling across the surface, but can't see what made them. Well, the fish are pretty abundant.
There. Yeah. Oh, yeah. They're just going across the same path. Nice. They're coming along at 23, which should be funny. They're catching up. It's a little bit. Yeah.
We're interested to see how far above the bottom the sea cucumbers go, and we saw our first on the way down at 617 meters, so something like 80 meters above the bottom. So they're not throughout the water, they just hang out near the bottom where I think they feed. Sometimes they go to the bottom to feed. Something's been disturbing the sediment there. I don't know what.
The grease comes full. And they grease the fitting that you, the straight one. No, it's not. But they're not in my position at the time. Tried to clean up the mess. Tried. The degree comes where it should jump. How'd that happen?
Did you tell them to move?
Yeah. They were moving. Oh, they're still going.
Going back and forth.
They're just going slow. Oh, you're right. They started moving.
Starfish, an asteroid, one of our first benthic invertebrates. There's something down here.
Go ahead.
We're just gonna go over to the west about 100 meters and then turn right back around if you wouldn't mind following us.
Go in the west and back east. Alex.
Think so.
The high concentration of suspended sediment is probably a combination of input from the river. Bio is a really big river. Also tidal resuspension. There's strong currents concentrated in the canyon. And then there's a lot of productivity in the surface that's also falling from below. But what keeps this resuspended, my guess, we're using this transect as an opportunity to take a look at the canyon axis and what lives on the central floor of the canyon. Look for a nice place to core. There's a small eel pout. That's the little curled fish. It looks eel-like. I don't know what species. A lot of rat tails.
You're gonna make it there position. Looks like it's slowed you down. Quite a lot.
They slow me down.
Yeah. It's fine. Just take your time. You go there.
Is that a crab?
They have 40 horsepower. I can unleash. Let's see if they let me be careful.
Oh, you won't launch.
Shrimp?
Bugs.
So when we come to the end of the transect, we'll stop and probably take a niskin sample and some cores. And then when we go back, we can just go to waypoint 2. We don't have to go back, recover our tracks or anything like that. So we can introduce all the people on the Science Watch. I'm Lisa Levin from Scripps Institution of Oceanography at UC San Diego. And we'll just let everybody go through and introduce themselves. Erin, you go first.
I'm Erin. I'm from the Marlowe Lab of Geomicrobiology at Boston University. I'm a grad student.
Good morning, everyone. I am Paola Santiago. I am part of the Cold Dark Ventus Lab from Dr. Turber at the University of California, Santa Barbara, and I am also a graduate student.
Buenos días, mi nombre es Praxedes Muñoz, de la Universidad Católica del Norte, académica en la Facultad de Ciencias del Mar.
Hi, I'm Scott Wyman with both the Chemical Sensors Lab and Clumped Isotope Geochemistry Labs at both Woods Hole Oceanographic Institution and at Massachusetts Institute for Technology, doing both some of my geochemistry work and then running the SAGE methane sensor on this cruise.
And we have, as you heard earlier, we have Tyler and Alex as our pilots. And Sophia, why don't you introduce, oh, I guess you did introduce yourself at the very beginning. Yeah. Okay, our communications expert, Sophia. How many more meters would you say we have?
About 10.
About 10.
Pretty sure none of them are
So we have no big surprises on this transect. It's sort of too bad we can't peer underground because I bet it is full of really big animals down there. Productive canyons usually have huge biomass of underground life in the sediments.
I don't know what it is.
A crab?
No.
Maybe a dead crab. Happy. Well, let's when to stop.
That's it.
Okay, stop
Rich,
transect
would you
one.
like me to continue us?
Thank you.
Alright, I'll set us down.
So we can set up for a niskin before we pick up the sediment. Are you okay to
Yes,
cord
sir.
here?
Thanks,
Rusty?
Rusty.
Yeah, okay.
Yes, thanks, Rusty.
Or
I'm just gonna fix the rest of the point of direct, right? That's okay. You want to see it pointed right there? Yeah. I wasn't entirely close to it right there. I see. Alright, set that. Should be working. fired.
Good, Mr. Lynn. Great. So now we'd like 3 push cars. Oops.
Oh, okay. Just the old security. Fortune out. Okay. No. There we go. What do you want to do, buddy? It makes no difference watching the hard ones. Won't you? No, I don't. Oh, speed. We don't know the scale. What torch?
We never know how sticky the sediment will be, and how consolidated, and whether it'll stay in our tubes.
You want to do a test, Lisa, or do you want to just?
I think we'll put in 3 and keep our fingers crossed.
Got it.
Hmm. Guess we can't see what's happening inside. Does it feel pretty soft?
Yeah, just went right in.
You might need to go in a little further, since they've been sliding down on, what do you think, Patchy, a little deeper?
Yes.
Yeah. That's good.
Perfect. Yeah.
Okay. Yep. Push cars are one of the deep sea sampling technologies that hasn't changed a lot in my lifetime. As far as I know, we were more or less doing the same thing with them 40 years ago, 35, 40 years ago. The quivers have changed a little bit, and they vary from vessel to vessel. Sometimes they have T-handles on the top, sometimes they have these bulbs, soft bulbs that you can see for the handles, and then the quivers that they go in. On this vessel, they've got both an inner and an outer quiver to help keep them from popping out when there's methane bubbles. But the basic technology of a tube with a handle hasn't changed a whole lot. The pilots make this look easy, but we know it's not. If any of the scientists tried to do this, there'd be a big mess. Okay, that's 3. If we get a picture, maybe we can zoom in and.
Got it. Thank you.
Good?
Yep.
All right. Oops. What you got? Let's get the more job one. Yeah, that's just everything else. Hopefully this will be fun. Perfect. What's that?
We're taking fewer than we usually do in our cluster of push cores because, here, like the chief scientist has said, he's full up with cores. One of the things that happens is that scientists run out of sample containers fairly quickly towards the end of the cruise, sometimes, but sometimes mid-cruise, and start conserving those. Does it look like it's falling?
There it is. Oh, thanks. Maybe a little bit. Little plug at the bottom. Where? No. Must be very, very soft at the top, huh?
Yeah. It's pretty short.
Yeah, you want some more? I know. Yeah. Might want to answer it. Just your, in a little, yeah, in a little bit. I'm trying to get my fingers, but there you go. Nice.
I wonder if we should we push them in even further? If you think that would help before you cover it.
I can't. I haven't done that.
As long as it can still grab it.
They're pretty far in, but I don't know when. It's sliding down, right, as you pull it out.
I can't tell. You can't, I mean, I can only see as much.
Yep.
It's there. I mean, I can't push it further, but that's gonna be up to.
Maybe just a little. Okay. Happens.
Bristol.
Yeah, that seemed to help.
Oceanographic, awesome. It's a big old snake. Good. All right. You wanna look at it real quick? Ooh.
Oh my goodness.
Oh.
Oh, he didn't see us.
Oh.
Oh no.
Look out, buddy. You turn. All right. You turn.
Visitor. So it's one of the things that's interesting to note is that we've seen a cat shark and a skate already on this dive, and these are some of the animals that hang out at methane seeps. But we know we're not at a methane seep. As best as we can tell, we're in a canyon, and so what it means is that the seeps are visited by background species that live elsewhere, and they are opportunistically probably using food, obtaining food from the seep sites. Means the seeps aren't really isolated from the surrounding deep sea communities. Well, that looks really good. You guys think we should take one more?
Yeah.
Try for another gym course. Number 7 was very short. Yes. Great. Oh, another. Is that the same guy? With something's on its tail. So could we get one more, please? Somewhere relatively undisturbed. Find a spot. Is that a shadow or a ledge right here? Just
It's a shadow from the
shadow.
water.
Yeah.
But see,
Yeah, it's just a shadow. It's pretty flat there.
Alright, let's put this in relation to the other hole.
Sure. Maybe there's a seep up here.
That would be exciting.
I don't think there is, but, you know.
We're good to grab this one?
Yes.
Can we just poke a hole big enough so we don't have any?
It's like counting.
On the side of, we need a lot of this at the ground before.
Good luck.
I don't know if it's really loud. Sounds good.
Very nice, thank you. That last one was much deeper than the short one, so we're good. Okay, so I think we'll just head back over to waypoint 2 along the bottom. Scott, you listed in the Stephen chat.
Sure. Also just saw something really cool on the boat chassis right now.
Is there nothing where
No, it's just from yesterday. So,
the methane readings?
yeah, I think for yesterday.
There was some other one.
On the bottom, I wonder if this is coming out of the estuary or sea, where it's slightly up. And it is a little bit elevated, similarly here to where it's here, closer to where it's there. So, I'm actually really excited about this.
Do you want to bring your computer over here so you can track it, or you want to show me something? Nice.
Yes, we can tell parents for this. They might be good noises.
So I'm gonna let Scott Wyman sit in the lead seat for a while and talk to you folks about methane as we make our way over to waypoint 2. He's in charge of the SAGE sensor, which is measuring methane on the ROV. And we've got a lot of methane aficionados on this leg of our expedition.
Go ahead. Moving cities.
What's up, Tyler? Hello, hello. They gave me the front chair.
Well,
Hello, Soph.
I thought Scott deserved to have a little lead time.
On the sedimented canyon floor, all of the things to collect and sample.
Talk about the
Yeah,
Talk
well,
about Sage.
talk about Sage.
Welcome back, Sage Scott.
Sage Scott is here, and Sage is working well, even in the canyons. We're not purposefully hunting for methane here. It would be wonderful if we saw some, but Sage is mostly on board for us to measure methane in plumes and on the seafloor at Seeps, but we've kept it on for our canyon dives this time around in case it could indicate to us if we come across seepage sites. I was just playing around with the data from yesterday, and it looks like the canyon floor here is a bit elevated in methane, which then decreases as we go up. Very, very small amounts, a couple of microatmosphere difference between being in the canyon floor and rising up the slope. So I think that might be related to our delightfully sedimented water coming out of the estuary, carrying all of the biological activity with it. But I'm excited to see if we see a similar pattern from today's dive.
Let's let it settle out.
Scott, what is the coolest thing you're seeing right now?
The coolest thing that I am seeing right now on the screen. Currently, sediments, which actually, as a geologist, are exciting. No, it's funny because this is what carves these canyons, right? The same current that's coming out of the river and carrying all these sediments is why we have these big canyons that we're all interested in transiting and exploring the biology and geochemistry of. So kind of part and parcel of having a canyon to explore is having water full of sediments to carve it. Ooh. We all just saw a sea cucumber go by on our downward facing. The gasps that none of. So Tyler, are we climbing to waypoint 2?
Yes.
Yeah.
Straight.
Cool. Awesome.
What's up? We did white mountains.
Yeah.
We have like 5 steps behind this conversation. No. Sorry, I'm just busy. Just trying to do this. Going over here. Trying to get Scott more sediment to look at.
Ah, great. This is when they put a geologist behind the mic, is when the biology is hidden by sediments.
Yeah.
No, yesterday as we climbed the canyon walls, we gained visibility pretty quickly, if I remember right. So hopefully that will happen here again, and we'll be able to do some biological sampling, some more video transects. These canyons kind of have a twofold purpose in these dives. We're both comparing them, we're doing video transects, so the scientists who are coming on the second half of this cruise can compare the canyons up north here to those south of Puerto Montt, where they will be doing their work, and then also comparing the fauna and conditions within the canyons at a similar depth to the seeps that we've been finding, and seeing the differences between our kind of biological communities at each place. There's a question in the chat about the source of the methane. On different dives, we've had different sources. So we've had both a number of canyon dives along the Chilean coast, and then a number of methane seep specific dives. Within the canyons, this canyon is the one that we have full methane measurements for. This, to the extent that there is elevated methane, which is very, very low, it is likely related to the water flowing out of the estuary region of very, very high biological activity that's only, I think, a few kilometers east of us. We're up pretty high in the canyon. Ooh. At the Seeps sites, oh, and a crab coming towards it too. Oh, sediment cloud.
Talk about sediments.
They're just trying to give me more sediments to look at, clearly. But just getting back to that methane question real quick, on the seep sites, we don't know the source of the methane. So one of the goals of this expedition is to understand where methane at different locations is coming from. At our first seepage area, it was very diffuse seepage. It was all throughout the area. It was likely shallow seepage that may be biogenically sourced. So most methane is split into either biogenic or thermogenic methane, which you can differentiate based on doing some measurements of that methane, particularly carbon isotopes. And I do not believe we were able to capture any gas at that site up at El Quisco. But that seemed to be from the geology of the area, relatively shallow methane. And then these Concepcion seeps, which we were at until weather drove us kind of a bit closer to shore, are located above a series of fractures and faults kind of between Concepcion and the Chilean Trench. And while that doesn't mean that the source of the methane is deeper, there are many more bubble plumes that may be fault or fracture related for transporting the methane up through the sediments. We did capture a gas-tight bottle of methane at the Concepcion Seeps, so some folks will be doing, I believe, isotopic work to characterize what the source of that methane might be. Starting to get into crab territory.
We're starting to climb a little bit too.
Yeah.
Just gonna try to keep it going. Until I'm perpendicular. Cool. That works for you. Just cruising. Should cross off this one. Oh yeah. They made back in the grid spots. They made superfluous. Yeah. And we should cross off this one. Up to date. Sure. What happens at waypoint 2, Scott?
That's a great question. I think the answer is we get higher up the canyon.
We just
I think Adam,
driving.
are these waypoints at Adam?
We have a series of waypoints
Yeah.
here that are designed to allow us to look at the south wall of the canyon, which we think is a little steeper than yesterday.
Yeah.
We'll be looking for different faunal communities, collecting corals if we see anything new that we haven't, or sponges that we haven't seen
See that
before.
closer to you.
But the waypoints themselves are not specific locations, right? Like when we've been diving on Seeps, we've been targeting locations that seem promising for specific faunal communities, whether that looks like carbonate in our ship's backscatter, whether it's mounds, whether it's bubble plumes. But for these canyon dives, I believe we've mostly just been doing spaced out
That's
100-meter
correct.
waypoints
We'll
climbing
try
the
to-
slope.
We'll be trying to do some video transects if
Yeah.
there's decent visibility to characterize the
This is too short.
Every time we go to the seafloor in a different place, we find something new and unexpected. Yesterday we had beautiful brachiopod and heteropolypus gardens and reef on the wall, the northern wall. We'll have to see what awaits on the southern canyon wall.
When we target canyons, as far as I've been able to see from a non-biologist perspective, we've been looking at finding kind of the steepest walls to transect and study. Could you speak to that a little bit, Lisa, as to why those steep walls are a really good target for exploring these communities?
Sure, I mean, usually the steeper the wall, if there's any hard substrate there, then there's less sediment cover, and you get a different kind of animal that will attach to a rock, a bare rock. If the wall is steep, then usually the currents flow more quickly over it, and it's really good for filter feeding animals. There are a lot of filter feeding animals in this region. There's a good starfish, the corals and sponges, and as well as the brachiopods and bryozoans that we've been seeing. So sometimes you'll get whole ledges which stick out into the current and they'll be covered with animals. In general, sediment cover tends not to favor dense assemblages of filter feeders. What is that?
What?
Need a science poke.
Well, it's maybe
It at
zoom
least needs a science
sensor.
look.
Oh, the cloud, here comes the cloud.
Oh, sediments.
Pink sphere. Oh, people were moving too fast for me to ask what it was.
Here's
So
one,
this is at
wait.
least the second we've seen.
Oh,
We can set here for a second.
it looks like Kirby.
Jigglypuff.
Oh.
Very true. Very trimmed forward.
I guess it doesn't matter, it's not about to be. Yeah, we'll just see what it does. And trimmed forward. So we can focus
Oh,
on this.
with a cat shark.
Let's see. I mean, it could be a mushroom, you know. Closed up heteropolypus,
Lots of Furby.
but maybe not. Are we as zoomed in as we can? There. Oh, cool. It
What?
is an irregular sea urchin, probably. Huh.
Whoa.
These are burrowing sea urchins that can live 10, 15, or 20 centimeters below the surface of the sediment, and they come up once in a while. You can see them on the surface there, but I think we got one yesterday, I think by accident. It came up attached to under a rock or something like that. It's the same one we saw yesterday. That's interesting. So they are one of the things that's underground when I talk about a lot of bigger animals underground. Irregular urchins are one of the common canyon and continental slower species.
You getting this page up?
Okay, we can continue.
Is this lighting good for you?
Amazing. If you wanna top down a notch.
We're talking to our EDC representatives here about the lighting conditions.
Oh,
That's a chimera.
Not bad. Interesting
whoa.
fish.
Come back.
So we're a little shallower now, we're at 674 meters. So we come up about 25 meters in depth from the bottom. And we are seeing a bigger diversity of things here. What else have we got down there? Some shells.
These shells?
They're shells.
Yeah.
These look
And a
like
skate.
Skate on the back.
skate. Yeah. Ooh,
I see
clamshells. What do you think, Scott? What kind of clams? I don't know.
Those are pretty big ones too.
Maybe it's shells. Nice skate.
Pedro, this looks like the one from yesterday, but without the marks. Doesn't have those marks on its wings.
It doesn't or it has?
No, not as deep at least.
But that one looks smaller
What's
as
in
well,
the
right?
tail
Yeah.
part of the skate?
Maybe that's like
for
something they get when they
animals,
go to the outer space.
Like a rite of passage. Marks up here.
What's right under us?
Could you zoom in on its tail, please? Just want to see if those are
It's very challenging for the autofocus in this kind of clarity watercolor.
Oh, but look at that focus on the sediments.
Anything you don't want to look at.
No, I know what you want.
This is what you want. No. I didn't touch it.
Fish, all facing the same direction.
That's so long.
Guys, keep your eyes peeled in case the clams shells become more abundant. And you know what we'll find.
We're not stopping at waypoint trip, we're just cruising.
I believe so.
Yeah, I think we'll, at waypoint 2, we'll come onto the track and
then
move
just
towards
continue?
3. Yeah.
Drift into it.
One of the things, if we do find seeps in canyons, they often sit at the base of the wall. Floor, but right at the edge. Before the wall starts up. Well, the seepage is probably a result of currents. Somehow the connection.
This is, that's that one.
Allows
that
them
one's
to
one.
so it sticks on.
How would you think? How would you guys focus?
What I do when it's
The
like
current
this,
is strong.
I keep it, I focus on whatever I am, and then I
Well,
shift,
that's what
switch
I'm
to
wondering,
angle,
what
because
these
then the
clams are,
stuff
and
flies.
whether
So
they
that's
are
what-
seep clams or some other.
Oh, you guys think that's what? Yeah, but when we zoom in, we focus.
Yeah.
And then you are stuck with the angle focus, which is super slow.
Yeah, yeah.
Tyler, if we see one of those large clam shells,
So I would have
oh,
that shell right. or
Yeah.
something. Now it's like here.
I was gonna say, if we see a large clam shell on the surface, let's pause and take a good look at it.
Do you wanna?
Okay, is there anything we can do about?
Yeah.
You said nothing on stage.
Just so they're not grabs of focus, right?
Yeah, it's kind of like
That's, yeah.
sees if auto gets too close.
Anything is going down slightly.
So like usually.
Oh,
what
good.
happens is like,
That's
it's-
actually
It's like in case like this would be supposed to go just autofocus.
Yeah.
Could you zoom in on
Okay.
that
Can we,
white
Then
object?
yeah, Tyler, can we pause and zoom in over there once
But
we have
Any recommendations?
a cloud?
if you wanted to play with it, if you could figure something out.
No, what I'm gonna do, that's the name of the camera, so this is a 17.
Yeah.
Could be another sea urchin.
Spreadsheets of it to see.
You can fix it.
No, no.
You can fix us.
I think like the ISO, so I think there's a lower ISO for that.
Yeah.
It's 200 or
No, we can go down once.
Someone asked for
Urchin test?
find out what's the
Someone
base
asked
ISO.
for two different
Because every
buckets.
camera has a base, what we call base ISO, which is the ISO where the camera works, the image quality is the best. Usually in older cameras, it's a
So
lower
that's
ISO.
what the irregular urchin looks like when it's dead and there's no spines. So that is what's called the
Frame
test
ISO too, 'cause I guess it's the,
skeleton of this animal.
You have a
the-
nice
something that I think you have to go into the meaning.
Thank you.
Thanks,
The
Tyler.
only other rule that we follow is if it's a 30 fps.
They're called irregular urchins because they're not fully round and spherical. Kind of a little bit
Oh,
horizontal.
you know what we're streaming at? 30.
I don't know what that camera is at.
That one, Lisa, that one looks like it's a little asymmetric too. Is that something that irregular urchins are? Okay.
You guys give me a 30, 25 maybe. I think it's 30. Yeah, I think 30 is completely right.
Yeah, maybe what we've been seeing is clamshells have been urchin tests.
Yep, that's quite likely. Good call.
Should be just kind of parallel.
Although that
That-
one looks like a clam
Tyler,
shell.
can we zoom in on that? Or pause and
Yes. Yeah. Cool. I'll just walk it out.
There's also
Is there? I guess I'm not walking it in.
Oh, I was saying the clam cells are just so sporadic and solitary that
They've fallen down from above, possibly. Or, I mean, it's definitely, looks like
That's a clamshell.
clamshell. Yep. But not necessarily, it doesn't come by a seep clam.
Yeah.
Okay.
Nice. Thank you, Tyler. Yep.
Yeah, I got from spawning on the same day. You go, I didn't even check it. She gave me these options work, and I said, yes, that option works. There was other one. I wasn't under the impression I could change it. I should probably check it.
If we're 100 meters from the next waypoint, we can do another video.
Yeah. Start one here as the visibility's getting a little better.
What do you think?
Hey, Alex, yeah, so you measure the distance to waypoint three
Yeah.
per floor. It's 135 meters.
Yeah. We could do one starting here and then end it 30 meters before
Yeah.
the next waypoint. Yeah. Yeah, so why
We'll be
don't
swapping
we...
pilots in about 4 minutes too. So
Okay,
just
so
be that
maybe
little
we'll...
pause.
Or we can also just wait for a pilot swap.
I'm gonna do 100 from there.
And then do 100 from there. I think that makes more sense.
Yeah, if you want uninterrupted.
Yeah.
It's just hard to keep in place.
Yeah.
In these conditions.
That makes sense.
Unless I'm gonna be right to work. Thanks for finishing that.
Many more eel pouts here today.
Looks like I've come up to a wall.
Yeah, look at your sonar shadow.
More steepness. Look up just a little bit.
Oh.
All these cat sharks, when we do find some hard substrate, we should be looking for shark egg cases. They usually require a hard surface or a biotic surface to attach their egg cases. So the corals often have them, the rocks sometimes have them, seeps sometimes have them.
Here's some stuff.
There is a lot of stuff. Whoa.
Yes.
Speaking
Good
of a
thing we
new
didn't start
surprise.
a video transfer.
What's the port mesh?
Beautiful brachiopod
Don't
and
need any
heteropolypus
Well, you built it.
alcyonid garden.
Wow.
We slow down and take a look.
Oh, yeah. Sorry, can you just-
Lovely.
Can we try another light that has less backscatter?
We need the center catholics is what we need.
that'd be on.
It didn't work. We can start turning off lights.
You wanna put out your,
We could do it here. Are you gonna
If-
sample here?
not necessarily, but if you want.
We could do a flyby, yeah, with our.
Is that better?
Wait, sorry, these aren't the
Yeah,
last piece.
yeah, it actually is. But the
Wow.
problem for me is the top line on the right.
Turning off the catholics made it better. Just remember that for my control.
Okay, I need to write it down. Just try to get the lights for it. That would
Is that- are
nice.
these
For
the
little
last
Charlie?
piece, or-
Yeah, but the problem is like
Heteropolypus.
it's too much.
Wait, little
But
Charlie.
Anthomastus is another,
What?
yeah,
Yeah,
they're
no,
really,
one little
they're
trout.
alcyonid.
We're gonna see with this visibility. Is that our
That's-
problem?
Pedro's problem.
Yeah, I'm not sure about this. We're gonna see. It's just gonna be quite little back center, but because of this red thing, they
Zoom in on that.
Oh.
Pink.
Oh, look what you did. That.
Yeah, a lot of pink worm.
Yeah, I think it's too much still for the
Okay,
truck.
okay.
It's too much still. If we can
Can
find
we
a
zoom
place.
in on that pink worm that's on the worm-shaped creature?
Lower left. Yeah.
Please.
Do you wanna? Are you gonna fly it?
Also, Scott, you gotta check the
Let me know if you wanna.
Falcor chat to see who wants what.
Yes.
I'm gonna check them down.
I haven't seen anything on that yet, but oh, now Amanda comes in. Orange bivalve.
Anna wants the orange part.
Yeah, if you wanna.
Greg is so disappointed that it's a cucumber.
Are you too close to this event? Because the lights are pretty high compared to the orange.
And they
That's
want
a good
the
thing.
acesta, which is the orange bivalve, which you'll not see right now.
I
Let's
did
do
see it
cucumber.
before the cloud came up.
Let the center is clear. Definitely a sea cucumber.
Oh.
There's an orange one up above, just up above this heterophones.
I was gonna stop looking at that camera. No, I was bringing it because you were swinging left because I was looking left. So as you were swinging left, I was centering.
Oh, that's all now we're right up. See lots of little tiny bryozoans as well as the big ones.
Oh, Greg would like the pink sea cucumber.
You see the orange bivalves?
Are those those?
I think.
There were bigger. I saw bigger
Actually,
things
I don't
before.
think, I'm not sure these are them.
Yeah, I think there's a different one.
There should be a frilly acesta.
That's what I saw.
A little bit curved on here, Sonny.
Mm-hmm.
So the sea cucumber, I think we could section.
Yeah.
Brought the lights down. Is that better or worse?
No, it's actually better.
I mean,
My
I
problem
can
is
take
the light that you guys have on the
Where you
top right.
have them.
Yeah.
That one is too close for my camera. I don't know which one is that.
I don't really have things.
Yeah, when that is off, all the other lights work. It's just that one that doesn't work. When
Do you wanna
there's backs together.
Slurp this cucumber? Is that what I'm hearing?
Yeah, we're slipping that cucumber.
Do you want to fly it or land it?
What do you want, Sonny? Do you want to put it in or do you want me to set it down?
Set it down. I want you to do this collection and then
There, Lisa. There's the
Can I set up?
acesta.
Yeah, I'm gonna kick both rams out because we got one
Get the
stuff
ram.
flying around.
Gonna give you a look for a light.
I'm changing the pilot watch right now.
Look at the rock box.
Is it just full of silk?
Yeah.
Nice.
Scott, keep your eye on the sea cucumbers.
Yep.
You know where to go back.
Pedro's impressed. Shot. The sections in which also
Alright, that's amazing. I want to get too close here.
Look at this camera study. Didn't know that play.
you only keep on your hand.
Only grabs stuff like that.
Scott, the sea cucumber needs to go in one of the course sections.
In the
5,
course one?
5 through
Okay.
8.
Awesome. Thank you.
So we save the fine ones for small, really small.
Got it. Or for like suctioning sediment type of stuff. Yeah.
when you
So
have
for
an
those
analysis,
watching, we have a section
find that
sampler with
diagram.
I think 8 compartments, and 5 of them don't have a coarse mesh
Oh,
on
you're so
them,
fine.
and
Yeah.
4 of them
There's
have
plenty of them.
sub-millimeter mesh sizes, so decide what goes where.
Are those are the same ones, right,
Is it?
Lisa? These are the same cucumbers, yes?
Yeah.
Yeah.
Looks like Kaniki,
Great.
Guys, any of those guys? I just felt like you. Where's the camera pointing? Up.
That was the call from the chat.
Right
Yep.
there.
Green,
I see him.
Greg, and Firm. We have a lot of the scientists on the ship watching the same image as the live stream from different parts of the ship, from the labs, from the galley, from their rooms, and so those of us in the control room are getting
Yeah.
messages on WhatsApp from
Great,
the
Jeremy.
various scientists who want different things collected, even though they might not be on watch. Try to look at
Yeah,
it.
go ahead.
Would you mind pausing the move right there?
Yep, I'm all stopped.
Oh, thanks. Sorry, so I'll just dump some. Work. You want jar 5, right?
Yeah, jar 5 would be great.
Start. Hmm? Alright, that's us on jar 5.
Awesome.
Oh, way to blow this is, Alex. Where'd all that come from?
Animals tend to look a lot bigger when we're viewing them through these zoomed in cameras, but the suction sampler helps us see how small they really are. I think the diameter of that sampler we're looking at now is probably about 7
Movement in the
centimeters.
jar.
So the sea cucumbers
Oh.
probably only
Sorry, did I move
about
the
2 centimeters across.
You're on bottom. But he said here, so you have to do a little square. I know. I was trying to do it with a little bit of visibility because of the, giant red thing on the end means you can't see what you're doing.
I'm gonna let go.
So, I see so. What'd you hear? Would you come up for it? Straight to. I don't like too much with it. The hose is a little bit short, so I'm worried about kinking. Like, see how hard the bend pulls now that we took the meter up a bit? Yeah. Oh, yeah, that's why. Because we've got a bungee and a super short hose now. Check the section up and stuff. Yeah, I'll ram in. You're not touching anything now. That's the one I was target fixing on. That's what you're looking at? Yeah. You can really turn it on and flush this one out. You resting on the seat? Alright, ran in.
So you know to fill this out. Don't do it till we get the sample.
I have put a time down. These are all right there.
Are we going up the canyon or today? Yeah.
It's, I think it's the same time of the
See,
night.
that's one of the challenges.
We will be going
Feels very Independence Day.
It's fine.
Oh. Like those. Oh, there we go.
It's in, suction off. Indexing away. That's suction off.
And then we also have one other sampling request here.
It's not indexing the other sheet. We're in the correct page. I assume so. Is that what we're looking at? We should index even a section.
Abigorgona sepala, which is the basket star.
You want some random
Basket
analysis?
star, did you see that?
Trying to be an all-star.
The corals.
Okay. No red.
Basket star, it's Abigorgona sepala. Maybe. I thought I saw one up on the other coral, but
Yeah.
maybe it's a different coral.
There's one down here too, right at the bottom of the. Oh, we're panning out from it.
That's not on, is it?
So let's see if we can see our orange bivalves.
Not here.
This would be a cesta. You should look really and orange.
No,
So I think
they're not these guys.
Let's do this one.
Sorry, Dad.
Sweet. If you think about it, you should grab the gear and like that. Yes. Yeah. Yes, I got this little video transect, which is just low, standard, rate of speed. It's outside the current, high visibility. All this is good.
Yeah, can we loop around?
Thank you.
We're on, we have one more collection request, so if we loop back a little bit to look at kind of the face of this, I think we'll be able to find what we're looking for. Think is a heavy word in this sediment, but I think.
What exactly it is you looking for? What exactly are you looking for?
Hold on, I'm hearing too many things in my ear.
What exactly are you looking for?
So there's they kind of look like these, but bigger. They're kind of an orangey bivalve with frills.
And it says they're next to the yellow octocorals. So
Oh.
maybe if you zoom
I
out,
guess maybe if you
find the
zoom
yellow
out,
coral.
which is right above. Oh, is that them? Could you zoom in there?
That looks like an acesta. Let's put it and see if this is the one that was requested. Was it Greg who requested it, or Anna?
Anna requested it.
I'd zoom up to that one above it, the bigger one. There's one to the right, actually. Can you zoom in on that and see if that's what they want?
That one you mean?
Yeah. It looks frilly, maybe. There's also a desmophyllum possibly next to it, or a caryophylla, can't tell.
That is the smooth film. This
Yeah.
one, very nice, but we don't collect.
No, but do you want the bivalve? Is
Yeah.
that the right bivalve or
No,
is that
it's
the wrong
orange.
one? Wrong one?
I think they're better ones. I think
They
they're
say-
lower down.
Yeah, because of.
There. That one.
Yeah, maybe
Right.
that one. Yes.
Yeah.
Yes, this one. This one. But you need to. Yeah.
Yeah.
Where?
That
In
one?
the haze.
Here. This one.
Yeah. Oh, nice focus.
Okay.
Thank you.
But that smooth film was very cute.
It's gonna be hard to get. Like, the question is, how will you pick it up?
We.
Yeah, there was a bigger.
with the
If we go
Ah, look, there is another one.
down off the front, I feel like there was some further down.
I saw a couple.
Yeah.
Oh, 2 this smooth film, 3 this smooth film. That's full. That only I can see more.
Down and to the right, there were a few, I think.
Here.
If that's them, that would be an easier cluster. I probably should suction them.
We collected
I know.
the manipulator yesterday.
I don't think that's them.
But I don't know if.
Just do it all up.
No.
Yeah.
That's not
But the
them.
moment I
No.
saw that one. I saw other, but I think we moved.
Yeah, Sonny, could we
Because I
reposition
saw a couple.
a little bit kind of lower down
Yeah.
that way, which is the way we came in on this little reef?
Yeah. No.
Because, yeah, I saw a cluster of 4 or 5.
Yeah.
Yeah. I had no idea what they were, but they were orange.
But nice. Big population. Big population.
It's a shame the visibility's not better because it's such a beautiful site.
Yeah.
Feels like
I should have sit down for a second and the second I think. Maybe it was because we moved a lot of someone.
Yeah. Looks like sunset. Everything goes red.
I guess some of these portray the end of times.
Ah, there we go. Is that-
No. Those not in here. Close to the.
Oh, yeah. That's a big one.
Vector coral. Yeah.
Yeah.
That one. Why is it good? Yes.
You see it? Oh, yeah.
Is this one? No.
Did we get one yesterday?
Yeah, just to the right of it.
I remember collecting one.
Hmm?
We got one yesterday.
Yeah, but we need another one because the
Mhm.
museum and then one for genetics. So.
All the time.
But yes,
That is one.
it's weird. No, this looks like.
Okay.
If it pops off, we'll probably still be able to grab it.
Sure.
It looks animated.
Cool.
Hopefully it closes.
No, it's similar, right?
I have no
I can't
idea.
see it.
It's very tentacly.
Let's see when we close. If they close like this,
Yeah.
here, because Because the other one is. There's a seep.
It's interesting how convergent the bivalves and the sea anemones are regarding tentacles.
Yeah.
See it on that one there, but you can't really see the color of it. T4
Yeah,
camera.
this is.
Yeah.
Well, let's see
You want
how
that?
it's
Up
close.
there.
Yeah. But
Yeah.
it's very.
If you touch it,
Yeah, Alice,
it
you
should
gently
close
poke it.
the valves.
Give it a little science poke.
Let's see if it's closing.
Yeah.
There's a.
Yep.
Yeah, it's a
Yep.
Bible.
It's
It's a
pretty
Bible.
funny when the scientist sitting in the van can't tell if it's a sea anemone or a
I'm
bivalve.
sorry, but there was too many tentacles. Looks
Yeah.
like.
So you want it?
Yeah. Yeah, thanks. You.
I want it.
Thanks.
Where are we putting it?
Cover or box preference?
Could I? The last time we put
Okay,
it yesterday,
coral
the
quiver,
coral quiver,
please.
because the
Yeah.
body box is
Yeah.
rocks.
Yep. Oh.
Oh, oh, oh. Hello.
What
Oh, look it. There are
That's,
many.
what is
another,
that?
Another here, another
Yeah.
here.
That's some sort of shark. That's not a cat shark.
Maybe a dog shark.
Yeah.
You want to open?
Do we need a niskin here, Anna, for any
I
reason?
will close it some neatly.
What?
I
We,
will close it
we
some
close
neatly.
one of
Not right here.
We close one at the bottom. where
Ah,
the
no,
cord.
okay. Here, yes, you
Okay.
can.
Yeah.
I don't know which one we are, but yeah.
Do, going, we're
Ah, okay. I think we are in the south
30,
walls.
30 meters off the south wall.
Okay.
Yeah.
Yeah, we close it.
Maybe 50 meters up.
Towards the end.
Before we do it.
Yeah, it's a Bible.
Yeah.
But it was
Very tentacle bivalve.
It's probably a good idea to close the
The camera,
niskin
you
first
already have a
because
keeper.
this could create more suspended
That's why I put the
sediment.
T4 camera
with the manipulation. Because you have some before with the.
What do you think, Scott?
Yeah, I mean, I was almost thinking if we didn't close one now, it would give the sediment that we've stirred up a little more time to settle.
But
I'm not
we're gonna
sure.
stir up
We
more.
can, no, we can close after. First the Bible and then we close it.
Okay.
So for those looking, the orange creature that we're trying to get right now is a true bivalve in phylum, whereas the rest of the creatures that look like they all have two valves are actually brachiopods, or the majority of them, and they're in a separate phylum called Brachiopoda.
Wow.
Can we zoom in on, get a nice picture of it? That's a good look at the bivalve and its shape before we
Yeah.
crush it.
Oh, the shrimp.
Thank you.
Alright, we'll play. pushing the thing on.
I don't know, but I don't wanna grab a bunch of them. I just have to grab a
Yeah.
Just hold on.
Oh, mudslide.
This is what I meant about the niskin. Oh,
Yeah, you got
nice.
it.
Nice snag.
Very nice.
Yeah.
Yeah.
Unless you actually, could you push it with that ramp?
Yeah.
This is going in number 1.
Yeah.
Yep.
Get all the way up. Yeah, that way you can see it a little better.
Let's use that camera. Use that camera. Let's look with the camera. Yeah.
Yep.
It's inside.
Yep.
Yeah.
Nice.
Well then, thank you.
Nice, Alex.
Now we can-
Yes.
It's interesting as we've climbed, the methane's been going down, like Scott said.
Scott will be happy to hear that the humidity is also going down.
Good. That is important for sage.
Okay. Anything else?
One niskin.
On this thing?
Yep. Which will be niskin 2.
You good?
So they want more acesta. 3 acesta for Eulogio and Javier.
We'll get those after the niskin.
So I've displaced Pachi.
And do you have to resume out of biological signal? I can write down niskin time and depth. I can swap out. I'm happy to.
Alright, Proctor,
Hello.
I'm gonna swipe back out with Scott.
No, it didn't fire.
Oh, you weren't doing one? Sorry.
No,
It was 2.
No.
Okay. You want me to put the niskin down here?
2 didn't fire. Lisa, 2 didn't fire.
It will eventually.
It will, but it's not yet. wing, I'm turning it back over to Dr. Lisa Levin to lead the continuation of this dive.
Collect a couple.
Of what?
The orange hideouts, like
The one that Alice just grabbed.
There?
Cheers, yours.
These are so boring. You somehow sent a voice memo to the group chat.
There's one.
1, 2, 3,
I sent a voice memo.
Yeah.
How did that happen? I don't know. My phone has a mind of its own.
potentially 4 right there.
1, 2,
3 and
3. Where's the other one?
4. 1, 2, 3, 4.
Oh, 4.
So how do you want them in the coral pavilion? Number 1.
They can go back in. Have you closed number 1?
Could open that.
Yeah. They can all go in there if
Yeah.
they think they'll fit.
Alright, reopen number 1.
Just a question of what's the easiest way to get them. Like knock them off and then pick them up, or use the scoop, or I don't know. Whatever Jed thinks is
Honor and pleasure.
gonna work. That worked. You can find them. How do I delete the voice memo?
I think if you hold it, it'll have the option to delete.
What?
I think if you hold it, it'll give you the option to delete it.
Thank you.
Number 2.
Great. I don't think that's it. I didn't see what. Did anybody see where it went?
It's down there.
Okay.
Could you reach that?
All the way down there? Wonder what's on it. looks like a limpet or something else living on it. Yeah. Let me see one more.
Yeah, there's 2 more of them, I think.
Oh yeah. I like the under ledges.
What? Yeah, there's one there and one there. Trace. Yeah, close. Close, 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 close. Yeah, I think you got it down.
Nope. Where'd it go?
Down there.
All the way down there again. The simplest task gives you appreciation for picking things up on outer space. It's a little bit the same here. Difficult. These are pretty fragile animals. Oh, down it goes. Maybe we just try a different one.
There's one more right there.
There's also another one right here, so we need it. Oh, that one broke. See this one? There. And there's maybe one here also. Which this one's probably easier to reach. But
Bit more. Yeah. There we go.
okay, so there should be four of these 5 ounces, right, total? I don't know if they're acesta. Do we, Anna and Greg, do we think these are a type of acesta? These orange ones, or something different?
Is that it?
Yes, thank you, so we can close it. These bivalves will go into the museum in Chile, and some will, I think, be used for genetic studies. So they are in the service of science. And it's often very important to establish baseline understanding of what lives in a place. So a lot of the collections here are the first for the Chilean deep canyons. And so scientists 50 or 100 years from now will be able to know whether different species have gone extinct or changed their distributions in response to climate change. So having these collections and having the new species described when they are new species is very important for our ability to detect change.
You happy for us to
Okay, we're
check
good.
that?
Yes, thank you.
You happy then?
Yeah, I'm just gonna wait till you move the rail so that I can look down the way.
Okay.
We could, how far are we from the next waypoint? It's more than 100 meters, right? We're at
Roughly
waypoint 2
100 meters,
now.
yeah.
Can we tell if it's all rocky? I'm wondering whether we could do a video transect here, just to try to capture some of this for the canyon people who are going to be quantifying things. And it would be really nice to be able to do it over some of the oh, there's a sponge. I wouldn't be surprised if Anna wants us to collect sponge. But, ooh, and a gaggle of sea cucumbers. Tell us if you think we could start a video transect.
Oh, yeah, you can
Yeah,
start
you can
them.
start it now.
Okay, start of transect 2. There's some more orange bivalves. We're starting to pick up a couple other things. I see a swiftia, some sponges. Bryozoans. The sponge is getting more abundant. Sea urchins, basket star, beautiful. Well, that's interesting. Getting some rock with more sponge. can see every time there's a slightly more vertical face with hard substrate, we get an abundance of life. If we weren't doing a transect, quantitative transect, I'd stop and poke around here and see what's on the rocks, but we'll have to do that when we finish.
So what are the white tube things?
It's a sponge, probably. Looks to me like sponge. It's interesting. There is a methanotrophic sponge that looks a little bit like that. But lots of Gorgonocephala, which are the basket stars. They're very elaborate ophiuroids. And there's a lot of them here, including some babies, little ones, which we don't see so often. Great filter feeding habitat. You can see everybody's facing the same direction. Somebody in the chat asked me why I spoke of a gaggle of sea cucumbers, and in truth, we don't know what a group of sea cucumbers is called. We could have an open contest for naming groups of sea cucumbers. They tend not to be very social, so we don't usually find them in large groups.
I looked into it, Lisa, and it looks like the group of sea cucumbers is called pickle.
A what?
Pickle.
A pickle of sea cucumbers. Is that real, or you made that up
No,
just
no,
now?
this is from the Natural History Museum.
Oh, that's
Source.
great.
If Paolo made it up, it deserves to be called that anyway.
Yeah.
So I misspoke. We did not see a gaggle of sea cucumbers. We saw a pickle of sea cucumbers. So we're on a video transect, which is why we're not zooming and panning the camera left and right and so on. We're trying to do something quantitative for our canyon colleagues. We'll be quantifying the fauna here. There's a lot of Gorgonocephalus. Tell us when we've gone 100 meters.
Yeah, we're almost halfway right now.
So, yeah. For those who don't know, we're off central Chile, off of Concepcion. There was a really, really big earthquake here in 2010. I believe it was an 8.8 magnitude. So this region was subject to a lot of shaking. I happened to be on a research cruise further south at the Chile Triple Junction at that time. We had about half the scientists on board were Chilean. It was a pretty major event to be out at sea during a huge earthquake that affected all the people on land. I don't think we know how disturbed the canyon walls were during that earthquake, but presumably there were some turbidity flows and excess sediment deposition. For those ecologists out there, we often look at animals and try to figure out what their limiting resource is, and you can see that for those species that live on hard substrates, the limiting resource is often just substrate and a place to attach. Sometimes there's 100% cover on the bare rocks, cover of organisms. I feel sorry for the people who have to count the animals in this video transect. There's a lot of them.
Between this and the first transect, it'll average out.
Yeah, that's true. There's a pickle of sea cucumbers down there. And now a lot of burrows. That big anemone is one that we saw at the Concepcion near some of the other seep sites we've been visiting. Maybe it was El Quisco. And we're picking up crinoids now. These are feather stars, I think is the common name, and they're the stalkless variety. They can swim and move around if they want to, detach. Oh yeah, there's an octopus, but we're not supposed to pan around or zoom, so we've got to just keep going till we've done our 100 meters.
It's not halfway there.
Hopefully there'll be some good stuff at the end that we can stop and look at. I think Greg wants a crinoid after we're done with the video transect, so there should be more. The murky water, the high particle concentrations we think are a result of input from the river, as well as tides. Biobío is a really huge river. Is our methane getting less, Scott?
It's kind of staying steady. It declined a bit, and it's close to a baseline now, which is similar to what it did yesterday looking at the data.
What is the baseline here?
Baseline right now for the instrument's about 9 to 10 microatmospheres. It's a little bit above that. That's not the true baseline. That's the lower limit of detection instrument noise.
Somebody in the chat asked if the water is brackish because we're close to the river, but the answer is no. Our salinity is 34.3, so that's a good full ocean salinity for the Pacific. So let me ask you, Scott, because you said there's 9 to 10 microatmospheres and that's the baseline. You think that methane is everywhere or just coming from the river?
Oh no, that's the noise level in the instrument.
Oh.
So we can make measurements of differences smaller than that, because the absorption peak can grow
Oh,
up
look
now
at
with
all
the
these
noise.
crinoids.
But the limit of detection right now with how much we've been sampling on this cruise is about 9 to 10 microatmospheres. We've gone up to almost 20, so a difference of 10 on this dive. No.
So I would say this is the biggest crinoid grouping I've seen so far on this cruise. Anybody want to tell me what a group of crinoids would be called? A congregation of crinoids, a compilation of crinoids.
I think we need to make one.
Okay, I'm open for suggestions. And we do want to collect these, but like I said, we have to get to the end of the transect. If for some reason we don't see any at the end, I suppose we could come back here and get these, get some, but.
Apparently they're called sea lilies as a common name, so I would pitch a bouquet of crinoids.
Greg.
Bouquet of crinoids.
Greg
That's
has.
great. He said he thinks it's the genus is Florimetra. A flock of Florimetra sounds okay.
That'd be good.
But flock is for birds. Sea lilies would be a bouquet, I think. Although you kind of have to gather them up to have a bouquet, right? If they were in the manipulator, they would be a bouquet.
Sebastian comes back up holding the bouquet.
There's really a lot of them, though, along with the Gorgonocephala. They're a beautiful combination of sea life. How far are we now?
25 meters from the end.
25?
Yeah.
The waypoints aren't 100 meters apart, though, so
Is that right?
have we been given, I think we just need to go 10 grid spaces. 1, 2, 3, 4, 5, 6. I feel like we've gone 10, haven't we?
Yeah, I think,
Yeah,
I
we've
think
gone 10.
this pair was 135, so if we're at 25 meters
Okay,
from the end,
stop,
that would be 100.
stop transect and collect. Well, can we go back down and get some, or there's
Oh, there we
some
go.
up ahead. That's great. Okay, so that's the end of transect 2. Yeah, these aren't, the waypoints aren't the transects. We're just going 10 grid spaces. So down here I can see, in addition to the crinoids, we have sponge, bryozoan, stalked bryozoans, ophiuroids, maybe an orange acesta, corals. Is that coming from us?
Suction for these?
Well, you tell me, what do you think? I
they
know
fly away?
they'd like them undisturbed, so it's fine to use a biobox space for them.
Are they gonna fly away as soon as I touch it?
They might swim away.
Yeah, that's what I mean.
Let's do a science poke and see what happens. I'm not sure whether these are avid swimmers or not. You could try to grab one and see what it does.
It's not gonna move.
Okay. Not moving. Sit on a rock.
Yeah.
You can pick up the rock if that's easier.
Falling down the hill in a second.
Oh, I got a shrimp too.
I guess we could get a zoomed in photo. Nice. Got it? Great. Which biobox are you thinking? They're all open. 4? So this is in 4B.
ROV bridge.
Go ahead, Tony.
Hey mate, at some suitable time it would be good if I could bring my head around to starboard bit, around like maybe 30 to 40 degrees.
trying to figure out how that's gonna work for us, Tony. Is it possible to go the other way? We're going uphill to the south.
Okay, I mean, if that's the case, I can just run on 2 engines and use a bit more power on it.
Okay, yeah, we're going shallower, so I'd rather not work in towards the vessel if you catch my drift.
Yeah, copy that. We'll keep it on 2 for now then, and then if there is any issues, maybe yeah, I don't suppose there'll be any issues, but we can always swing round and put us north of you and have the weather on the stern. But yeah, we'll stick on 2 for now.
Roger that, thanks.
Can we get one more?
Yeah.
Oh, actually, Greg just said one of those. So we can not collect another one. We don't want to take what's not needed, so. Unless, yeah. Such beautiful sea life down here. But I feel like we're on the dune planet with sandstorms periodically. What's the name of that planet again? Beautiful.
Just head them back towards the line, right? If you see anything you want, just head, I got pulled away from the line, so I'm heading back towards the line, right?
Okay.
If you see anything you want on the way,
Very well. We're happy to just look. You sort of get the feeling that if there were more rocks out here, there'd be more crinoids out here. There's like one on every rock. I wonder if they're territorial. Bigger rocks have more crinoids. Interestingly, the crinoids don't have a lot of commensal life living on them. We often see other animals living on the corals. Sponges are very clean because they produce natural products or secondary metabolites that often are anti-fouling agents. They basically keep other animals away. And that's usually why sponges look so pristine without other organisms living on them.
Brad, you got an updated waypoint?
Copy that.
We're getting back on our waypoint track. We were pulled off a little bit, so we're just coming on to waypoint 3. There are, I believe, on the order of 10 waypoints, so maybe we'll be only a third of the way up the slope. No, maybe 9 waypoints.
20 meters away from the wall.
We're now at 567 meters. So we've come up about 150 meters or so off the base of the canyon floor. I think visibility is a little bit improved up here, not a great deal. Does it seem better to you guys than when we were started this morning?
I don't know whether we
Just get sideways and crab across. As long as you can get the waypoints that go up the hill and go across. You gotta stay closer to the wall.
Is there a current pretty, are we being pulled by current or by
No.
the ship or?
Yeah, the current from the west.
Could we zoom in on the burrows for just a minute? They look a little different to me, and I want to see if there's any features that are notable. They don't look as smooth and round. They look kind of like the sides are dug up. They're much more irregular than what we were seeing before. You can see some, I don't know if those are tubes on the sides or just, they look much more cavernous, like holes in the bottom, rather than nice cylindrical burrows. So maybe there's something down there with an underground tunnel system, and these are just the surface manifestations. Okay, thank you. There's a lot of them, clearly, so something's under there tunneling around. Goodness, that's a really big crinoid down there. I wonder if we should just home in on one of these rocks and try to get some really nice imagery. Is this a good one, do you think? We can do some close-ups. Maybe we can pan around the rock and then try to zoom in on a few of the different kinds of animals on here, just to record them.
Just look around like this. No.
We have crinoids, corals, anemones, sponges, ophiuroids, galatheic crab. What else? Sea cucumber. Arborescent foraminifera, agglutinating forams. Brachiopod. Those might not be galatheids. Little Caryophylla coral, cup coral. I don't know if those are style crabs, types that hang out on corals. Maybe a desmophyllum even. Could we zoom in on this, please?
Which one? Yes, I'm sorry.
Right here. It could be a desmophyllum. Yeah, we don't often see that. So it's kind of neat. Grab a picture of it. Oh. Crab's on the move. It's kind of a hairy crab. Certainly got a lot of spines with things attached to it. Well, it's interesting. It's not causing the coral to close. So the coral doesn't seem to care. You can see the polyps out on the orange coral next to it also. Corals are really, we call them filter feeders, but they're also microcarnivores because they have stinging cells in their tentacles, and so they're able to capture very tiny swimming creatures, like the little, you can see a few little swimming plankters around here. Crab's probably wondering what all that light's about. Okay, that's good. Let's take a look over on the other side. Ooh, there's all sorts of fecal casts right outside that burrow, so we know some deposit feeders down there making the spaghetti-like sediment strings. I don't see who it is. Could be a burrowing cucumber. Let's take a look at, so those are brachiopods. Let's look at what's underneath this crinoid. It almost looks like a soulless cucumber, which are these scaly sea cucumbers that are flat on the other side. Not sure if that's what that is, but it might be. Interesting. The crinoids look busy. Not sure if they're feeding way down there at the base of their mouth. Okay, so Anna said she actually would like a small piece of sponge, but unless we have a big sponge available on this rock, we'll look for a different spot. I don't I suppose we could try to pick up a piece like that, but we might find some more easier to grab ones without disturbing everything. I don't-
Greg suggests that the cyanopticals are full of eggs, or is it? Oh, Greg wrote something in the chat about the
Greg said the crinoid pinnules are full of eggs and sperm, and the one we were just zooming in on. Forget which one that was already. This one? Oh, yes. If you see the pinnules are what makes these look feathery, and you can see the whitish part have the gametes, eggs and sperm. Like on this one right here. Yeah. Nice. Presumably they're released out into the water for fertilization. Okay, thank you.
Did you want to sample anything or?
So I'm gonna suggest that we look for a place where the sponge is more prominent and we don't have to disturb all the other creatures to get it. So we can continue on our waypoint. That was just a little photographic interlude to check out the organisms, which are so beautiful. So that might be a good sponge to collect. We just need a piece of it. And it could probably go in a coral quiver. We're diving here on this part of the canyon closer to shore because there is apparently really rough weather offshore. It's hard to know because it's really nice and stable on this ship where we are. We're in the lee of an island, and so it's very protected. And we're lucky that we've been able to dive every day. It's been interesting, this 50 meters below us or so maybe 100 meters down, but they haven't been everywhere on the canyon wall. Don't know if they have narrow depth ranges or need specific current conditions. Maybe it's too muddy for them down deeper, too much suspended sediment. John showing us where we are now on our trek and where we are off Concepcion in central Chile. I have fond memories of Concepcion. I did a sabbatical there in 1998. Where I met Hachi.
What?
I said I met you in 1998.
Oh, yeah.
Yeah.
So far.
Fun fact, right? Do we have an open quiver? No.
Yeah.
Do you know which number it is?
5.
5. Okay. Oopo, go back in. No. I think you got a piece of it in, right?
Yeah, a large piece.
Okay, that's good. That's fine. You don't have to worry about the other.
I think so.
Yeah, right down there, yeah.
Somebody in the chat's asking whether it's warm here, but we are really close to shore and we are pretty shallow at 557 meters, so it's kind of a normal temperature. We're in temperate latitudes. Thank you. So, coral quiver 5. The sponge. So far, the coral quivers have a bivalve and a sponge, but no corals. We should just call them quivers. Yeah, thank you. Actually, yeah, Anna's probably very appreciative to have a picture of how this sponge looks on the rock. It looks strange. Looks like shapes out of some Dr. Seuss story. Blobs. This rock also looks like it might have a desmophyllum and crinoids, anemones, brachiopods. You can see how clean the sponge is. Not too many things are living on it. It looks like the crinoid that's hanging on there is on a something attached to the sponge. Oh, there's an isopod. I think it's an isopod. Yep. These are small paracarid crustaceans. Well, the crinoid's on the sponge. It's just got a lot of aggregated sediment there, mucus. Looks precarious. There's another acesta, the orange bivalves. Pretty. Very nice view. Oop, there's another little isopod on the sponge above. Isopods are related to roly-polies, basically, the little pill bugs that live in your garden. It's the same group. Oh, here's a bigger one, I think. These guys?
To the right. Right there. That one?
Is that an isopod too?
Looking at
This
it.
one? Right here?
Yeah.
Very cool. I guess it's the same thing as this one. That's the back end and that's the
Yeah.
front end. The big dark eyes. Hmm. I don't see anybody requesting that we sample them, so I guess we can continue. Beautiful community. Rocks are fascinating. They're home to many great creatures.
Lisa, what do you think those are?
These?
Yeah. The translucent thing.
Oh, well, I thought they were brachiopods, but no. I don't know what those are. I can't tell if they're hard or soft.
Science book.
We can poke them.
So,
I think they're little soft blobs. I mean, they could be tunicates of some sort. They almost look like colonial tunicates. Could we zoom in, like, right there? Are there a bunch of little individuals all glommed together?
I'll almost zoom in.
Okay. I think everybody else is eating lunch, so we're not getting any input from the rest of the science on board. But that would be my guess, is some kind of colonial tunicate. Okay. There's a chitin over there on the side. That's a nice looking one. Chitins are mollusks with a radula that scrapes food off from below, from underneath them. They also have a big foot. And there's some branched bryozoans down near the bottom here. These white fan-shaped creatures, I think, are colonial also bryozoans. There's isopod, another isopod, all sorts of creatures on this rock. I bet if we sampled this rock, we would find 100 species on here. don't think that's a worm. I think it's a feather star that's attached to something. There are stalked ones.
Would that be an opioid or an asteroid then?
The chitin? Yeah. There we go. There's a big polykey tube probably right behind the feather star arm. And of course, there's probably all kinds of small polychaetes in amongst the mud. So, okay, great. I think we can continue. Just give people an appreciation for how diverse the community is on these rocks.
Moving on.
Yes, please. We can just continue along the waypoint towards waypoint 4. But slowly, in case we see anything really cool. It's important to remember while we can see all the life on the rocks, we can't zoom in and look under the sediments. I'd love it for somebody to invent a camera that can look through the mud and show us all the animals down there. People do it with sediment cores. They put them in X-ray machines and so on and can see a lot, but it's still really hard.
Red RV, you got an updated waypoint?
Copy that.
Can pan over here and just
Yeah.
see if there's anything. Oh, beautiful. Something new and green down there, a green sponge. You always have to wonder what motivates colors in these animals since it's dark and they aren't seeing color. Why are they so colorful? Oh, it's not a sponge. Looks like it's something colonial. Maybe a tunicate.
Okay, good to know.
That would be my guess. It's a colonial tunicate. Hmm.
Would it be a Solante please, or they usually not?
What?
A Solante, or do they usually not?
The Solante.
Sorry, Solante. With a C.
Maybe. I just don't know that, I've never seen them share a home like that, but maybe they are, I'm not sure. There's a really big inhalant siphon
Yeah.
right there,
Uh-huh.
which makes me think tunicate, but I'm not sure. They've got little tiny creatures on them, little white spots. I don't know what those are. They're copepods or?
That's as far in as I can
Okay. Ostracods almost. They look like ostracods actually, sitting on the surface. Benthic ostracods. Okay, thank you. Gotta look. Definitely not a sponge. At least as far as I know. But it's a really big flytrap anemone there. Another nice rock. Good thing they're too big to collect. Or I'd want to bring it home. I wonder if Anna wants this is clearly a different sponge, the elongate one. I don't know if she wants a piece of that. Couple of different species. But I'm not hearing any requests, so we're not gonna collect it. We'll just continue. So pretty. Get a screenshot. So we've only done 2 transects today, is that right? Yeah. Are you guys gonna change out in 10 minutes, in a few minutes?
Yeah.
So we probably shouldn't start one now. Yeah.
Brad, you're already got an update waypoint.
Copy that.
Almost vertical here. It's funny, so the crinoids seem to have filled the role that Brazingid starfish might in deeper waters, or maybe more clear waters, hanging off the ledges. Lots of acestus here. We could have got a handful of the orange bivalves here. Oh yeah, we can zoom in on that maybe. I haven't seen so many octopus here, not as many as at the Seeps, but they're clearly around. Wonder if it eats brachiopods. An Arrakis moment. We have to hope that the animals that live down here are used to having periods where there's lots of suspended sediments around them and it won't hurt them too much. I think canyon walls are inherently unstable and subject to high turbidity. Sort of interesting to look at the particles that are up in the water and see how many of them are actually living organisms swimming around versus how many of them are suspended sediments. Quite a few of these little suspended particles we're looking at are actually plankton swimming. You see how everything's swimming around. Thank you, Doug.
I'll just come over here, right? See if I can just
So we are at a depth where we might be getting vertical migrators coming down here as well. But probably make a lot of food for the filter feeders.
Wes Anderson palette.
So we had stopped to take a look at this octopus.
Have you seen that trend of like if Wes Anderson the standards they're applying, they're like, oh, you can do that at sea, but it's just like cottage. It already exists. Yeah, we famously already made that already. My first one did one for like a day in the salt marsh.
A little bit of sediment in the water.
It's a little sediment covered now. But hopefully it's not gonna hurt him. Or
Really fine.
thank you.
Ready for this? Let's have a lock on. So, the line isn't in line with the hill, right? So the hill is, undergrowing, it's on the left hand side. So I'm facing the hill there right now. So you're kind of crabbing along the. One of the push doors quarantine. We didn't get back number 9. No. Like before we died, I went and checked out for one failed.
Try to fix the rubber first before you.
I just told somebody has to read the room. No, there's a test. They'll fix it. Yeah, we'll take the dragon out. Or stop anyone in. Number 9. Took one of these new ones. He's here. So still have one. But, and we didn't do anything much for they did jump out.
How about you?
Right. You good?
That's a
Morning, Greg.
how's Ben? How's everyone?
Looks a little bit dusty down here, doesn't it?
Yeah, good rest,
Morning.
Morning, Meg.
Buongiorno.
Buongiorno. Buongiorno.
How's it going, Greg?
Good. I'm excited to be here in this canyon. Hi everybody. We're on the afternoon shift and heading up the south wall of the Concepcion Bio Canyon, just off Concepcion at 540 meters.
Just arriving.
We're going to keep on going. I think we could do a transect. We're due for one. Just a meter or two off with the lasers on, and 100 meters. And tell us when you're ready, and we'll log it.
All right.
we're not zooming or stopping. We'll just keep going.
Yeah. Alright, here we go. Turn direction of movement, yeah. You wanna follow the line, I take it? Gotcha.
Not yet.
No, so you start the first
They're
thing.
getting ready.
I do the first thing,
Yes.
transect.
Yeah, we'll do a transect.
Okay.
So, for those of you who haven't been listening before, this is the first leg of Falcor 2's Canyon Seeps and Vents of Chile expedition. We're obviously in a canyon today. We've been in Seeps, but the weather forced us in to this remarkable canyon. We've been at kind of the shallow end today and yesterday. Hopefully we'll get a chance to dive deeper. And we may not have said goodbye to the Seeps of Concepcion. That's a decision that'll come later. And then we've still got about a week more on this cruise before we hand over to the next team.
So, my left side is now on the ground. I'm trying to get you as close to the ground so you can actually see what's on the ground. But just to give you an idea, this is the visibility situation.
Maybe it's not a good idea to do a transect right now.
You know what I mean?
Yeah.
Like, if I fly by, you're not gonna be able to see much. Does that make sense?
Yep.
So like I'm on the ground right now, I can't see the ground very well. Does that make sense? I'm concerned if I just do a transect at my minimum altitude, you won't be able to see anything. I can try to tilt it now, but like this is what you're working with here. You know what I mean?
Because
of the visibility.
the viz and also the slope of the canyon right now isn't suiting us. Okay.
Yeah.
Let's just go up a little bit, see if it gets better.
Yeah.
So we'll hold on the transect.
Well, this is what your transect would look like.
It's a very big canyon, and we're reasonably close to shore, so the turbidity here could be a number of factors. There's a lot of plankton, if you saw the beginning of the dive. And that all could be the marine snow you could see that could be contributing to the poor visibility, where there's a river runoff, and just a lot of sediment turbidity is also possible. I expect when we go out to 2,000 meters or so in this canyon, it'll be a lot clearer. But it's a good place to be a filter feeder, as we've seen. Going up the canyon wall, lots of sponges, a lot of sponges than we've seen anywhere else. Lots of crinoids, feather stars are filter feeders, and lots of octocorals too. So that cute eel pout. We've got a mercurid, rat tail. They've been friends with us on most of the dives who've done this leg. Okay, so now Levi's got us in position. We can see the green lasers 10 centimeters apart. Got a few clams there that look a bit like sea clams. This afternoon we've got Anna Adamo on watch with me, as well as Fernanda and Peter, who are logging. And we'll be with you for the next 3 or 4 hours. We collected push calls already at the bottom of the canyon, so if you're a push call fan, you're out of luck. No more today. After suction sampling maybe, and collecting a diversity of animals to go to the National Museum in Santiago, and for genetics and other purposes.
Bridge updated waypoint.
Update, thank you.
Morning, Eric.
Good morning.
This little hard rock. Those worms look
They're big sea cucumbers. Yeah, we got one of them earlier on.
Continue up this.
And we got a feather star earlier. That's the orange crinos. Yeah. And the white things are mostly brachiopods, which I don't think we've gotten today. We got a few yesterday. Levi, we might look at that if you haven't got too far. The pink thing there.
Right.
See what that is.
You give me partial zoom. Them on the bright laser there, the pink ball. Try to get you a little.
Oh, yeah, that one.
This one. Pink.
Oh.
Yeah.
Oh, it's
You want me to get closer? I can mark.
no, I think that's good.
Okay.
Greg, there is something here. This one?
Just hold. Anna's seeing the purple thing, dark purple.
This here. This one.
On the bottom.
Lower star looking one.
Oh,
down.
lower?
Yeah.
Yeah. No, wait.
Zoom in a little bit. Ben. Hold that to the bottom frame.
That one.
Centering now.
I couldn't see it.
Oh, I see.
Maybe you.
Hmm.
Can you zoom in?
Yeah.
I see it.
It is a sea star. Yeah, it's a slime.
We never saw it like
It's a
this.
slime star.
Ah.
Well spotted.
Oh, look at this.
Yeah, I think we can take that. It's interesting.
Might be some ram action there.
Thanks.
Zoom in. Somebody, the steering ship. Keep an eye on it.
It's pretty small. I think we could slope it up.
Can you point to it for me?
Down here.
Down there.
Sorry, there.
There.
There we go.
That's it. That's
That
central
thing.
there. Yeah.
Oh, yeah. Down right a little bit.
Right there.
Yeah.
It's only 2 centimeters. Good eyes, Anna.
No, thank you. It's because I didn't know what was. I was curious.
You might want to pivot a little bit. Pivot support.
Roger, ready for.
So we're on the wall of the canyon. It's pretty steep here. Presents a challenge for sampling. And the wall has a lot of brachiopods. Those are the white shells you can see. They're not a mollusk. It's a different group. As well as the shrimp, sea cucumbers, feather stars, and anemone. But we're after a little sea star.
Alright, I'm dry line, so
Yeah.
you should be able to get that section out. Which jar number would you like?
We could go into 6, please.
Roger.
Okay, it's 2.6. That's an image.
You're on 6. You want some camera? Down. draw a camera here too.
We feel pretty lucky to still be able to dive. We haven't lost a diving. But just where we are, it's still blowing 30 or so knots, and that's keeping us in a bit close to shore than we would prefer to be. But it's turned out to be a pretty interesting sight. The wind should start easing today. Maybe tomorrow we'll be in a different place. Remains to be seen. Maybe if we could try and get a zoom on it, Levi.
We'll do a zoom first, Ben.
Yeah. Yeah. I think it's what's called a slime star, juvenile, potentially. We'll get this grab off to sea star expert Chris Ma, see if he gives us a name. Okay, thank you.
Is it the only thing you need in there? You want to get the clamps?
Yeah, you could take a few other things there, thanks, Ben.
Ready for section?
Yeah.
There you go.
Okay.
Got it.
Keep sucking until the draw's a little bit cleaner. Pretty sure land though.
All right.
Nice.
Great.
Section's off.
So more than likely we're going to be using a suction again. So I'll just hold it for now.
Yeah, that's probably a good idea.
Okay.
Alright, so 6. I'm gonna mark it on the sheet.
Okay.
Alright. Ready for me to start running a little bit?
Good.
I'll take off and then pull my torque ram in.
Okay.
Alright, here we go.
Straighten the camera up.
Alright, let's work our way up this picture here.
Yesterday we were on the north wall of the canyon, which had much less slope, so it was much more sedimented, and we only saw a few rocky outcrops like this. Here it's much steeper, much more room for animals that require hard surfaces to hang on to.
Full feature, isn't it? Overhanging top.
Yeah.
We'll put you in manual focus. It's not looking so bad. That's cool. You're just deep here.
Yeah, they like to be out a little more. It's better for them to feed.
Just for your information, since we're so close to the wall, I set the range at 12 meter, 23.
Yeah, about sun, right?
Yeah. So that range is really 2 meters away.
Yeah.
batch of orange clams is they're called acesta. We collected some of those earlier. We collected another species in the other canyon at a deeper depth. So this is, I think, if it's anything like California, there's like kind of a deep sea or deeper sea acesta and a more shallow species. But neither of these we've collected here are the same as in California. And I don't think either of them has had their DNA sequence, so that will be interesting.
Thank you.
Oh yeah, you might want to take this.
No, zoom in.
Yeah, thanks. And then this is a proboscis of an anchor worm, so we could pan over that way.
Oh, no.
Oh, and another proboscis there. Yeah. So those tongue-like things are highly retractable. They go into a...
Which one are you looking at, Greg?
Oh, this thing.
Center white.
Thanks, Ben. The feathery.
Trying to keep it center.
It's a feathery sea cucumber. That's called a solid, P-S-O-L-I-D, and it's fixed in place and puts out its tentacles as feathery fronds, as opposed to most sea cucumbers, which crawl along and put their tentacles on the seafloor and pick up detritus. And solids tend to be plated, so they reflect in a way their sea urchin relatedness. They're kind of hard. And the
Fortunately, stay here. I have to like cross.
well, I
this.
think we'll collect that sea cucumber, please. You saw the proboscis is retracted of the spoonworms or tongue worms. Not really called tongue worms. Spoonworms they're called, or anchor worms is a common name. And they just go back inside, sometimes more than a meter, to a sac-like body. And they use that proboscis to feed on the surface of the sediment. The proboscis can fork in some cases, and that gives them the nickname anchor worm. It looks like an anchor and it's often green. Or in this case, it had a spoon shape at the end and it was white. If we see another one as we go on, we'll get a better grab of it. Now we're trying to stop and we'll see if we can pick up the solid, not sure the genus, filter feeding sea cucumber.
Tickle back in.
Let's wait to your right, Pete. Look at the sonar scan.
Is that it? No.
Go back to where to let it clear out. I want to get closer. I had my port ram out, so this time I could gently touch against my port ram. Can you see it right now? Is it below us?
It's below us.
Okay, I'm gonna go down even.
Yeah, I can't see it.
It's below that big white.
Okay.
They can pull the feathery thing in, so it might have retracted.
Slowly more. Another half meter.
Okay.
that it there?
Yeah, I think so. Hopefully this clears. Thanks for
Yeah, that's it.
This should. I'm up against the port ramp, but you know, I'm trying to figure out.
So
That's definitely it, right?
that's it.
Okay, good.
Yeah. So the solid's head is at the top with the feathery appendages coming out, and you can almost see the plates on the body, and then underneath, holding on with tube feet, just like a sea star.
There's another one right on top of it.
Oh, great. Yeah, we could get them both. So it's got plates a bit like a sea urchin, and they share an ancestry with sea urchins.
That one.
Nice. Well over 400 million years ago. But this platedness, which is reminiscent of a sea urchin, is secondary. So most sea cucumbers have little calcareous discs in their body wall to give them some support. And they're otherwise soft-bodied. But in this case, those discs have expanded out, so it's a convergence to look like a sea cucumber, a sea urchin rather, and to allow them stability as they filter feed a completely different lifestyle to other sea cucumbers. We could go to 7 for this one. Thank you. Yeah, the way they feed is the tentacles, they have 10 and they branch and they put them in their mouths one by one and lick them clean and then stick them back out. So it is kind of cool to watch them feeding if we had time, because it can be a while between each. This is a good time-lapse video.
Go for it.
I think it should clear. Hopefully this dust cloud clears. I think it's coming from below us, yeah.
Might wanna land and start without this. It's a little bit long.
Parked against the stubborn right now.
Here you are.
I'm parked against the port ring right now.
Yeah, just stick it out a little bit so it'll have a little bit more extension. Doesn't have to be all the way. Just give me as much budgie as possible.
Hopefully it won't extend it. Extend a little bit further.
So Scott, our methane master, is here. Scott, what's the story with the methane in this canyon?
Boom. There we go. I'm still putting it together, and I don't know until we go to a deeper site. But the dive yesterday and the dive today, we've seen actually measurable concentrations of methane down at the base of the canyon, which we've gotten a little bit higher levels today than we did yesterday. I think what we're seeing is we're getting methane export from the big estuary that is right kind of quote unquote upstream of us. The Biobío River, which is a few kilometers to our east, ends in a very large estuary, and estuaries are big producers and sources of methane to the environment. There's not much constraint on their kind of marine export of methane, so potentially seeing this transport is really interesting. If we do have a deeper canyon dive tomorrow and see less methane, I'll be really interested to put those transects together and see if we can understand a little bit about how far some of this kind of bottom water methane gets transported by the river.
Would it be like terrestrial debris, organic matter coming out and then
I'm not
breaking down into methane?
Sure, it's not something I've thought about before until making a depth plot of yesterday's dive. It could be terrestrial matter that is being broken down and is essentially being turned into methane actively in this environment, or it could be kind of very high concentrations of water methane that are being carried down within the water mass. I'm not sure which of the two it is, and we wouldn't really be able to pull that apart with SAGE.
Yeah, so in the canyon we were at the north, at 2,000 meters or so, we came across some interesting little mini seeps, and of course that'd be something we'd like to see when we dive deeper here. Although the source of that methane wasn't really clear either.
Yeah, and unfortunately I wasn't able to have SAGE on that dive, so we weren't measuring it within San Antonio Canyon.
Yeah, it's over the corner. What do you think, Ben? Think you got enough visibility to try it here? This one starts to see now. This might be your window here, Ben. Trying to focus. Okay. You try autofocus? Okay. Back in main autofocus. Go for it, man. Yeah.
solids have diversified into
Wait a sec.
quite a few
Here you
different
go.
kinds.
Here you go.
One of my favorites is called a sea apple,
Too low.
which is beautifully colored and lives on the reefs of the
There you
West
go.
Pacific. Purple and yellow and red. But most are usually this kind of color, pink or white.
Is that full size?
Yeah, it is now.
You might have to tap it, Ben. It's got little feet holding it onto the rock.
Got it.
Nice.
I can.
Is it sucked in one?
I know.
There it is.
Suction off. Let me know when you want suction again.
Wow. Still there.
Let me know when you want suck.
Right there.
Here we go.
Nope.
Yeah, a little bit. Yeah, you gotta go deeper, more away from the orbit. Yeah. Sucking a little bit. Deeper. Yeah, scrape it off.
There you go.
No. Drop down. It's there on the top of the anemone. You
Cover
see
the
it?
whole anemone surround it.
Yeah, it's just resting on the anemone there.
Oh, come in.
Yeah.
Oh, it got a little damaged.
Oh.
Oopsie.
I think it was attached to the rock. The
Oh,
whole rock.
hang on. Don't worry about it. Go back to where you were.
Okay.
And there's something poking out.
Keaton.
Section off. What do you? Yeah.
I'm looking at this. It's pulling back in. I don't think we'll be able to get it.
Alright, second
Try.
now.
I think it's a big polychaete. And there is a mollusk there that we could try for, the dinosaur.
That thing that retracted.
Yeah, that kind of shiny.
Yeah, it's still there.
Looks like it's rock. Yeah.
Yeah, it's wrong.
But Ben, just over here, behind the nozzle, if you retract a little bit,
Yeah.
this thing might be hard to get off as well. It'll be quite attached.
Alright, I'm second.
That's a chain of mail shell or a chitin. They have 8 plates or 8 shells. You might be familiar with them if you can get to the intertidal rocky shore. They're common there. But there's lots of deep sea ones as well, including this white one.
So it's attached to a small rock.
Yeah.
There you go. Okay.
Oh.
No.
Got it?
Not quite.
There.
Still there.
It looks good, but
They're pretty good at holding on, but
Oh, that.
got it.
Got it.
What a master.
I'm still sucking just to make sure it's in.
Okay,
See,
so
the shell is floating around in there. There you go. There's a bunch of.
yeah, I think it came through.
Alright. You want anything more while we're here?
What is that up there?
Yeah, no, we've got the brachiopods. I think we're good. We can continue.
Roger.
Thanks.
Alright.
Which job is that?
7. Alright, onward and upward we go.
is this looking for a transect now?
Yeah, we could try it.
Wanna
I'm-
try?
Like, it's what I'm noticing is the visibility comes and goes.
Ah.
Like the last, like all that dust that blew in, I don't think that was made by us. It's like it came, blew in, and blew away. So this, right now it looks like I could do a transect here, but I don't know what the future
Well,
has in store
yeah,
for us.
okay. Let's give it a start.
Okay.
So we'll start a transect 100 meters.
So just follow that line.
Yes, thanks.
Is this a good speed?
Perfect.
Usually. I'll just try to like give you the best view of the bottom.
Yeah, that's
Just
great.
To see the most things.
Ooh, it's an urchin I'd like, but hopefully we'll see another one. So we're doing transects repeatedly on the canyon dives that we do because the next cruise in particular, the next leg of this cruise, has much more of a focus on canyons, and they're heading further south, so the idea is to compare the canyons from where we were, San Antonio north, and then here, and then we'll do another canyon. This might be our last canyon. I think we go to another seep site after this before we finish. And then they'll be doing a series of canyon dives. And these transects help compare the different canyons in a quantifiable way. So we want to try and do 5 transects today. So we'll do one here at about 500 meters, then maybe another one in 100 meters up at least, and then again when we get to 300 meters.
Octopus.
So that's when the lasers at 10 centimeters become really handy.
The waypoint 5 is 9 meter.
While a lot of it then gets visually annotated with machine learning now, some of this hard work can be automated and the organisms can be identified and counted. And we've been at Scripps working on a project off California where lots and lots of ROV transects have been, after digital learning of the organisms, can then identify them and count them.
That's a big yellow.
That's a fluorescent sponge. I'm seeing a lot more of the little slime stars, so maybe they weren't juveniles after all.
Look at those colorful sponges on it.
Wow.
Yeah, that's
There.
a new sponge we're seeing. It's really encrusting. It'd be hard for us to sample, but if we see it after the transect, we should try. we'll expect to see a change in the community of animals as we go up. Here we're at 500 meters, it's pretty well oxygenated. If we get up to 300 to 200 meters, there'll be a dramatically lower oxygen level, so the community of animals there might be quite different. Ooh, that's a beauty.
Oh.
These rocks that project out really
Really angular too.
really accumulate lots of filter feeders. And we're starting to see brittle stars on the rock. Now that's something we haven't seen earlier. But it's really dominated by chrono. It's the most chronos we've seen on this cruise. And chronos are another echinoderm group. And the sponge is remarkable with its fluorescence there. But yeah, chronoids are echinoderms. Think of them as like a sea star on its back with its mouth up, and then the arms branch out. And these have on their back have these little cirri to hold onto the rock, but they can let go, and many can even swim. But when they're babies, they have a stalk, and their relatives in the deep sea, many of them continue to have a stalk as adults. So there was a transition from stalked forms to the feather stars, which break off the stalk when they reach maturity. So as babies, they have a stalk. And if we, sometimes they keep it as juveniles. So if we see one of those, we'll point it out. That's an anemone looks like we were seeing at the Seeps up in offshore, Concepcion seep, methane seep area, about 70 kilometers away. How do we estimate the distance traveled? Are those squares really the?
So that's X and Y distance, right?
Yeah.
Yeah.
But is it
So I
giving
started basically at waypoint 4.
Yep.
So he can measure me out 100 meters and then I can.
Okay.
400 meters away from waypoint 5.
Once we get,
Yeah, so we don't want 100, it's just 100 meters of ground traveled, which X and Y gives us.
yeah.
Near
I'm
enough.
traveling a lot because it's steeper.
Yeah.
Yeah, the number I'm looking at is just X and Y.
Mhm.
So if we travel 100 meters of X and Y, the slope would be more than 100 meters of actual ground.
Got it. Yeah.
Yeah, I'm trying. Am I doing okay? I'm trying to keep you close to the bottom and look at the good stuff here.
Oh, that's perfect.
We're doing okay.
It's
All right.
someone's going to have to review these, or a machine,
Yeah.
some kind of program, and just count and identify. So this is ideal, and then maybe measure things.
Yeah, I'm just trying to keep the biology and the.
I think you'll be doing a lot of these on the next leg. Are you on the next leg?
No.
Oh, you're off. Okay. You're on the next leg, Ben?
Yes, sir.
Yeah.
Yep.
Ben will be doing a lot of this.
There's my next slide. Great.
No, I'm leaving. A student of mine will be coming up. I'd like to be on the next leg. Triple junction could be very interesting indeed. I'll be watching. So the next leg, looking in late November, they'll be down at what's called the triple junction, where there's very likely some kind of hydrothermal vent activity and some potentially interesting animals.
Does sound good. Who's that?
Whoa.
Oh, it's another.
Oh my god, what is super.
Well, okay, we'll terminate the transect.
Terminated.
So this is another
Oh.
hook-armed squid, Gonatus
Yep.
onyx that the team saw the other day. With a brood of that blanket structure is basically a giant cocoon.
Kill lasers.
It doesn't have any eggs left.
Yeah, it looks like her eggs may have all hatched out.
Just as slow and steady as possible, please. That's great.
She's not able to swim very well because of that burden. And she's probably near the end of her life, actually.
I was thinking.
She's been holding that for months as those what would have been thousands of eggs developed and then have progressively hatched out. And you can't really see any of the
Talk to me,
embryos.
Pedro. What do you need?
Are you with the camera or are you driving?
I'm flying.
I was like, if whoever's with the camera can just like do closer shots to the eyes and the face.
That's Pedro from the BBC helping guide the shot. What we can see is
Yeah,
she's
that's great, Ben.
showing a lot of damage to her skin there. It's abraded and
There is no more.
decaying.
A
Oh,
few
they're still
there was
hatching
one.
out. So she still has some babies hatching out.
She's exhausted.
Yeah.
Really.
So it's not.
The eggs are much darker.
We could slurp a few of those little babies for genetics.
Go for eyeball. Go for eyeball. Go, 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 go. Zoom, 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 zoom.
I'm spotting.
There's lots of them coming
Yeah, I
off.
can go for the eye. if
Yeah,
possible.
yeah,
I
yeah.
couldn't.
Not my understanding
You
last
want me
time.
to focus?
Yeah.
Yeah, now they want to focus on the eyes.
Zoom out, zoom out, zoom out, zoom out.
It's getting shadows.
Give me lowers. Give me lowers. He's gonna get prop washed pretty soon.
I'm losing her in my camera. If we can turn the vehicle
Yeah.
to the left a little bit.
Yeah.
Yeah.
He's gonna get prop washed pretty soon here.
Could I see?
Oh.
Yeah.
There
Yeah, be
is
careful.
some small one para darle around. So we can start.
Yes, we would
Some.
like. Yeah.
Oh.
She's been on the bottom, it looks like. There's sediment all over her.
Oh, she's coming in.
Ah, look at how many she is.
Yeah.
If we close the niskin bottom.
Oh my gosh.
There's a lot of
Catch
babies
babies.
just got shed then.
Do you want some of that?
Yeah, we'd like to slurp some of those paralarvae.
Here,
No,
hold the
let's.
camera
Well,
while-
what do you think would be easier?
Just go for slurp. Start slurping.
Slurping.
Alright, where do you want me to go?
We just need one or two. We can sequence it and tell if it really is Gonatus onyx.
Yeah, that's oh, look at. Oh, that you can see it.
You're gonna have to fly it up.
Roger. What drawer number you want?
Put it with the fine mesh. So number one.
Look at that one.
Oh, there's that.
Back it up.
we love
Back
it.
it up.
She flew above me.
Back it up.
I think she flew above me.
There's a piece of her cocoon has come away. So we'll take that.
We got an index to jar 1,
We
yeah.
might even be able to sequence that, actually, rather than paralarvae. It's going down.
It's under?
They're collecting.
Oh, okay, okay.
Yeah.
Maybe.
I gotta get you to 1. I gotta get you to 1. Alright, suction's on. Here we go.
Is that too big?
It's in the lens. It's in the lens.
Maybe.
Back it up, back it up. There it is in sit. There it is in sit. Oh no.
That's right, Ben.
Wow.
Wow.
Ben Hirschon with the blues.
Wow. Thank you, Ben. That was great.
That was number 1. We're going.
Okay, I think we'll try for the DNA just from that. Do we
How we
want
can call it?
Did it come through? Oh yeah, there it is.
Come on down. Increase your range and so on.
Yeah, I got it raised up to 35.
Okay.
We'll get back to the wall here.
Another piece right there.
There you go.
How far do you think we went before we got distracted?
We can
close.
we can measure it.
Yeah, it's probably about 20 meters almost.
He's talking about transect.
Oh, transect?
We did
Oh,
that.
yeah.
Do you think we did 80 meters or something?
No,
We can measure it.
horizontal wise, 17 meters.
No, we started at 1.4.
Hmm?
We started at waypoint 4.
Oh,
On the
from
transect.
waypoint 4.
On the transect.
I thought from the line. Okay, stand by. From waypoint 4, we traveled 60 meters or so.
60? Yeah. So for that transect, we'll just call it 60 meters.
Okay. Alright.
Yeah, I made a note in the notes at the start.
We're done.
She was about to die. She was quite sick.
Yes. Yeah.
5 meters, so.
Camera center. Back on.
Yep.
Shrimp.
So we've used the name Gonatus onyx for that squid, which was originally described from California and is well known from the North Pacific. But here we are quite far south of that. So it will be useful to compare if we can get DNA out of that cocoon to compare it with DNA records further north to see if it really is the same species. Right, we're come up about 50 meters so far on this dive. Started to see some new things, brittle stars. I think it's a different kind of sea cucumber to what we saw earlier today. So I think we should try and get one of those.
Which one?
One of these things.
Right.
Thanks. Do you want
I want to see the next.
what?
I want to see if we also catch the larvae
Yeah.
tonight.
If you see another one, yeah. I think that would have been tricky.
Have you say that is different from the Gonatus antarcticus?
Alright, Ben, we're gonna go for a sea cucumber on the ground.
Three different
I'm gonna,
species?
I'm gonna try to get the port ram against the deck.
I don't know if Gonatus antarcticus is
Alright, got it extended partially. Take it in.
Might be tricky to get DNA out of that.
Maybe
thing.
the
But maybe there's paralabi in there, so. Are you interested in that green, yellow, the luminescent sponge?
sponges, yeah.
We're going after the cucumber, right?
Yes, please, Ben.
Alright, which jar would you like?
Just one. We can go to 8, please.
Alright, you're on 8. So Warren has business on it now? Okay, we're on 8.
You ready?
Yeah, I'm on jar 8. Ready for the sec? Alright, here we go. Not yet? Alright, it's off.
Okay.
Alright. I think your wrist is touching your tube is not far enough away from my kill section. You got a wrist roll. Forward.
Try that.
Okay. Alright, per second. I think it's behind the nozzle there.
There we go.
Okay, so.
Can you zoom in?
Yeah. Right here at top, section's on. I think it's past your nozzle.
Yeah. Hand up.
Yep. You can twist the wrist. It puts the nozzle away from you. Yeah. Bingo. Alright, she's off.
Don't turn it on.
Yeah, it's off.
Can't see anything.
Yeah, we'll let it clear. Should blow past. Slow viz makes it interesting. Bring some in on this.
So here you see a sea cucumber that's more of a classic form with a soft body. You'll see when
There you
the
go.
suction touches it that it's pretty much a collagenous body wall with a few little, well, not we can see very well, but these are very soft bodied and they have a ring of tentacles that they pick up sediment with. In contrast to the solid filter feeding sea cucumber we saw earlier in the dive.
I almost feel like I want to pick this thing up.
Still there. We can kind of see it.
I think there'll be plenty more if we want to get somewhere easier.
Okay.
There was another one on top, wasn't it?
Yeah, I think another one was nearby.
We could get this one. Is he sticking in the bottom? Right?
No, it wouldn't be very sticky, but there is one just up here.
Yeah, we could fly up here.
See that one? Sitting
Yeah.
out in the open.
We can reach on it at the moment.
Where is it?
It's up here.
It's just
See it
there,
sit?
unfortunately. Yeah, you can see it in the sit. Or we could fly on a little.
Yeah.
I think there are a lot of them.
Okay. Find a different spot. Copy. We're zoomed out. Hopefully we get some clearer water. Yeah, I think it's your best view of what you're doing. Yeah.
Touch that thing.
There's clear water above us. Take off and float up.
Yeah.
Hopefully there's some clear water.
Clearance. Okay, I think you're safe distance from the wall.
Yeah. Clear water, no water, clear water here. Let's see some stuff here. Alright, so I took this one out. Some things for you. Alright, we're looking for a sea cucumber for you. There's one there, down there. One
Yeah,
there.
there's lots. There's one there,
Go
there,
yeah.
up here.
That's one.
Maybe that's easy.
Here we go. I'm just gonna easy bring it in. I got my port arm all the way out to keep me like as far away from the wall as possible. Reduce. Alright, Ben, see the one we're looking for? See the one we're going for?
Yeah.
Alright, I'm gonna come in. So let's see. Bring it down, bring it in. Can you reach that from there? You can tilt here.
I think it's a long one, so maybe if you ram in, that's
get a zoom when you get convenient?
Yeah.
I'm gonna park here. You take my camera and zoom in on it.
Okay, that's great. Good enough.
Okay, go for suck. We're on jar 8. Alright, let me know when you're ready for suction to be enabled. Good sit.
Yeah, to ram out, starboard ram over.
Okay, so we're ready for now.
There you go.
Right, we're on jar 8. It's looking a lot better here. Here you go. You got suction. You want to get both of them?
No, just one's fine.
Just one, understood. Easy. Easy. I'm gonna give you some zoom so you can see what the little silicone's doing. Yep,
It's got,
gentle.
you can see the tube feet that it's holding on with.
Gentle.
Holding on pretty tight.
Real gentle. Those 2 feet.
Trying to find more.
Yeah. Good job.
There we go. There we go. You're doing good. Yeah. Wow, that thing really is holding on, isn't it?
It's mighty.
Hand up, somebody hand up. Zoom in. Can you operate this thing, Sophia? 1 minute. Zoom in. He came back out.
Did it,
No, it's still there.
It's still hanging on.
There.
It's still hanging on.
oh, I see. Yeah.
Can you zoom in?
It's different to the one we collected earlier this morning.
Scrape them off. Swimming in. Swimming in.
No, it's the whole rocks come off.
Whoa.
Hold on, hold on.
That is
That's
really
good, that's good. Can we bring the rock?
Sure.
Okay, we're going to buy box.
Okay. Good plan.
Okay.
I don't want this rock to get stuck in the
Yes.
console. Oh, he's strong.
Never seen a sea
Don't
cucumber
let go
do
to
that.
suction.
Understood. Which bad let's go 4A. Oh, 4. Right there. 4A. Do that.
Whichever.
Fortune.
It's impressive.
Wow.
Very impressive. Which one? B4?
4A is free.
Port side. You go more helpful.
Yeah.
Let me know when you want me to kill suction. Then you gotta dump it in. Maybe the weight of the rock will slide right in.
Here we go. Okay.
Alright, here we go. Section's off, so it's on you to wiggle it out.
Oh.
Nice. Oh, thank you.
It's okay.
Good job. Alright, so 8 still vacant.
This is anyways out because there are a lot of sediments. Because I put here, but the SS8, I
Yeah, we
think
might
it's...
still use it.
We are not going to use
We
it.
might.
Ah, okay.
The chamber is, we'll let most sediment through.
Okay.
You doing this? You'd like go over here?
You can have a look around. Sure.
Look at this. Alright.
What's this, Anna?
What? Ah, the one that we were
That's a
seeing
better
before.
version of that sponge.
Sponge, yeah.
That's,
It's weird.
it's very weird.
It looks like a dark green.
I mean, it looks like.
We should sample it.
Yeah. That in case
How big is that, Levi?
Let's get some lasers on it here.
I'm gonna start putting this one away. I'm gonna pick this up, Greg, right? If we're
it's
gonna pick
less
it up.
than a centimeter.
I think we might pick it up.
Roger.
Or
One
a
of the,
piece.
lasers go for the next one. Alright, do we have lasers on it yet? I can find my lasers. Oh, there they are. They're out there.
I don't know if it would break or come apart.
Okay, we'll stow the suction sampler and then we'll
Yeah.
reach down with the
Okay.
soft
I'm just gonna
draws.
drop this.
You want to stow it right there?
I'm gonna pick it up again anyway.
So we're going to try and sample this, what looks like a very unusual sponge. Unusual to us, at least.
There you go, man. Yeah, I think if we reach it with the soft draws, hopefully we can cut off the rock without damaging to that, maybe. I mean, it's pretty big. I think it's a firebox, not a quiver. What do you need, Ben? Want some light? The lows are already up. These are 8. It's mostly clean.
Yeah, I think we still might use it.
Okay, I'll stay on the
Close to.
Yep.
Still on this trap consistent sulfur lines.
Alright, so I might move the vehicle, but we might be able to reach it. Dark green, big ball. It's pretty far down here. You're gonna be careful with it.
Yeah.
And we're gonna carefully bring it up to a bio box. We don't know what its consistency is gonna be like.
We need to,
I can change heading starboard a little bit.
no, that's right, sit down a little bit.
Okay.
Just watch the shoulder.
Oh yeah. Yeah, you might be able to wrist left and
Yeah.
avoid number 8. Watch, shouldn't be a little bit.
Zoom out a little bit. That's good. It's actually kind of feels firm.
No, he's not touching you yet. He's above it still. Should be okay.
I think it's crumbling.
Looks squishy.
Hand up, hand up.
Yeah. Looks squishy and stuck to the rock. You have to like scrape it off. That looks pretty good.
Oh yeah.
I barely have it. Don't zoom out, don't zoom out. It's hey Greg.
Firebox?
Yeah, it's
Yeah.
gonna, it's like a sponge.
Yeah.
If I point it down, it's gonna fall.
Yep. So any of 1, 2, or 3.
Are you ready for your
You wanna
porch?
put it with the slug?
Yeah, go ahead.
Porching, porching, porching. And number 4 is open there. And go with the
We could put it in 4B or 4A, either one.
either one. Here.
So might as well put it with the slug.
Yeah.
Okay. Nice.
Tilt down. Tilt down. It might float off. Can you zoom in a little bit on the seep?
Yep.
You want to talk about eDNA and sponges?
Okay.
Yeah, we're just gonna drop it.
Good.
Wanna know? Right?
You're up against the divider.
They fall off?
No, it's not
Maybe
falling
once
off.
they
But you're up
close.
against the divider. Twist. Yep. And I up and away.
Great job, Ben. And Levi, thank you. Well, now Anna's going to tell us a little bit about why this sponge might be useful.
Yes, the sponges are
wanna get that
filtration.
cover lid right there?
They filter the water naturally because
Cover
it's
it
the
up.
way how
Yeah, that lid that's down here.
they collect their food, the nutrients.
I can't
So
really like
the
that.
idea is to use them as piece of part of the sponge for eDNA, for the DNA, environmental DNA. So in theory, we can use part of their tissue to see if there is DNA for other animals that are surrounding this habitat. So the idea also to compare this with the water sample that we collect so far and see if there is a comparison, a useful tools.
We'll watch it.
This is what we are
we are trying to do. Also because in some cases we don't have enough water to collect during the dive, so maybe this little sponge will help us a little bit more to characterize the habitat. And it has been used in the last years. There are a few papers where actually they explain how they use these sponges as an indicator for the eDNA, and it looks like that works very well. So this is a good opportunity to test it again in a different environment.
Onward and upward. We'll keep going up.
Yeah.
Can I-
Thanks, Leo. Yes.
Can you close the niskin?
Roger that.
Let's do a niskin before we take off, please.
Increase your ISO.
Sure.
And open iris. Okay, I'm going to show it through laser. We're going for 3.
Yeah. It's retracting, I see it. It's just going slow.
There goes the yellow tag. Fired.
Okay. Do I go further to the car?
No,
Let's see.
see.
Yeah. Okay. That's number 3.
Is 5, Greg? Is 5?
Yes.
I couldn't see the level.
It flipped.
It flipped? Sorry, I lost it.
At 5, right?
Yeah.
Happy?
Yeah, we saw that.
Yeah.
Okay.
So that'll give us a 5-liter water sample that'll be filtered later tonight. All right, let's maybe try transect again. What are the conditions looking like?
Let's see what we can see here.
Yeah.
Get you closer. Back to the line. That's the sea cucumber with a rock attached to it. Okay, can I dry it?
Close your eyes a little bit.
Alright. Heading back to direction of movement here. Here we go.
Okay, let's start another transect.
Octopus.
Here's 5.
Roger.
Okay, so we'll go through 5 and keep going.
the ground there a little bit. Just to give you an idea. Like, I'm trying to keep it as close as possible to the deck.
So that cloud's not us.
Right, we just swim into that.
Yeah.
Pretty brutal. And basically up against the wall. And I still can't see it. See it? See that? Just do that. I'm coming up easy. Floating up. I can slowly approach again now that I came up a couple meters. See if it's visibility better and I get back to the wall. There it is. Alright. It's something.
Bridge, updated position.
Update, thank you.
Eric, just to let you know, we're just moving small increments because we're in a wall with zero visibility.
Thanks for the info.
Roger that. It's really working.
Yeah. I'm basically like dragging my starboard ram is kind of what I'm doing right now. Like trying to get you as close as possible. But let's blow up a little bit.
There might be something in front of you.
Yeah, let's see that. See how it's basically it's normal to it now.
Yeah, you're gonna have to go up.
Okay. Come on.
It's a meter short.
Okay.
4 now. There you go. That's something.
I'm gonna try to go back in.
Okay.
No, leave it there. It's okay.
Okay.
Because I back off, I want to be able to see something.
Roger. Let's see what happens if I close the light.
Okay. Sorry, Greg.
Oh,
It's pretty bad visibility.
it's not easy, yeah. It's,
About 3 meters away from it.
Roger.
Yeah, no matter where you go, it's silty.
okay. Well, let's cancel this transaction.
Cancel?
Working, and we'll just move on up until we can get clearer water.
Let's see some.
Yep.
Wow, look how silty that is. It seems like it's all coming down.
Yeah, right. Yes. It's like a bit of a slide up above us.
Yeah.
About a meter away.
Roger, let's go a bit.
So bear with us, everyone, while we try and get some clearer water.
Hey Greg, can you move your mic a little bit closer?
Oh, I thought someone moved it away.
Not me.
Oh.
I think they move it away if they want the whole room, and then they have it close to you if they just want you.
I see. How's that, Julianne?
Yeah, it's good.
Okay.
There's a lot of, these are all crinoids, right?
Yeah, all
I've never
crinoids.
seen so many crinoids. This whole dive is just solid crinoids. It's kind of nice that because I can actually, that's the only thing I can see on the seabed right now.
Yeah, they shine through. Well, we've hit a bit of a silty patch. It looks like the wall's not so steep.
I think my port ram contact failed.
Yep.
It's hard for me to see.
Must be low tide.
So this is the second canyon we visited on this cruise.
That's all the crinoids are panned.
We did 2 dives in the San
Right,
Antonio
right.
Canyon. One at about 1,000 meters, another at 2,000 meters. And that'll be relevant for canyons further south, but also the 1,000-meter one was a good one for us to compare with the seep environments, which we sampled when we got up higher than 1,000 meters, shallower than 1,000 meters. Gives us an idea of what organisms are living at, say, 400 meters or 500 meters. And here we started off at 700 meters, and the seeps we were visiting earlier this week at Concepcion were at about that depth. Ideally, we would have started at 1,000 meters, but the weather stops us getting out deeper in the canyon right now. Maybe tomorrow we'll go out deeper, or who knows? We're still waiting on the wind.
Oh, crabs.
Yeah, there's quite a few crabs here.
Oh, we got our crab friends. Maybe they've been making all the
Yeah, the rock-
sediment.
Yeah, look it. It's have to be more.
See them all on my sit too, Greg?
Yeah, okay. So if you were watching previous dives in Concepcion methane seep area, we came across a huge aggregation of these crabs.
Wow.
Yeah.
And again yesterday. And these kind of spider crabs. Oh,
Just
that's
stay here,
a
keep
true
moving.
crab. It's a brachyuran actually. Well known for migrations.
Can you turn off the laser as much as possible?
Yes.
Thank you.
Are you interested in putting out your light?
So that's the thing. How steep are we?
It's very
Right.
steep.
Yeah.
I wouldn't suggest it. I think it's not enough viz.
Yeah.
Might lose it.
It's gonna fall.
Yeah.
Yeah.
Did we ever get a name on this crab? Anybody?
Pressure still.
If they're just, let's wait for the other crabs to arrive. It's like a war scene.
Roger. Holding this.
Yeah. Thank you.
Which camera are you using, Pedro? You want our camera or are you shooting yours right now?
I'm using mine, but
Okay.
if you guys can do some close-ups, yeah,
Roger.
like that bend, that looks great.
Alright, we'll focus on the zooms.
Sophie.
I think you got some sediment coming in around here, guys.
Yeah, I can see it rolling.
Yeah, it's rolling
Yeah, let it come and then we good.
It's like a sandstorm.
in. Yeah.
Is it high tide, low tide, or what? You know?
There's like.
Okay, that's. I'm going here. Do my show.
Oh, is there too much sediment, Pedro, or?
Yeah, no. Can you make more?
Yeah, yeah.
No, I'm good.
Okay. Alright, we can zoom out. Keep
Thanks,
going.
guys.
Yeah.
Zoom out.
Alright, let's float on here. Onward and upward and forward.
Yeah. We're not sure where all this silt is coming from. Could be an army of crabs a bit above us.
Yeah, it's almost deep low tide.
Yeah. That's why it's all coming down.
Roger.
What's that?
Lunch, probably.
Yeah.
I think it was about this depth yesterday when you found the crabs.
440.
You guys want to leave a mark there in case you guys want
Yeah, would be nice.
Sure.
Love crabs.
Are you still interested in our footage, Pedro, or can we put the lasers back on?
Oh, you can put the laser back. Thank
Okay.
you.
to? Lasers on.
Alright, I'll keep going.
So Maritza, one of our macrofauna experts, suggests this is Libidoclea, the crab. Thanks, Maritza.
He's holding up his lunch too. Hello. Bye. You just see the reflection of these glowing arms and crinoids. That's the only reason I know
Yeah,
my
it's handy.
proximity to the wall, really, visually.
Yeah, so Libidoclea granaria is a margid crab. Libidoclea. And they've been one of the highlights of this cruise multiple times with their candy striped legs, large aggregations. They're pretty aggressive. Margids are often called spider crabs. They're a true crab. You can see they've got 4 walking legs and 2 claws, as opposed to the lithodid king crabs with 3 walking legs and 2 claws.
Reach up the position.
Updating here.
We saw this luminous green sponge earlier, so we'll keep an eye out for that. We have a look at that. Maybe we'd want that sea star as well, but we'll see how things settle.
You want to grab the arm before the lens.
You're going to zoom in. Okay. Ben, on the green sponge. Okay, go.
Suction.
Suction.
If it's hard, yeah, we can try.
If you get some pieces, it's good.
Alright, Ben, I want to select the green sponge. I'm gonna get you parked.
Got it in your sit?
It's just a thin film.
Okay, starboard ramp should be pretty much all the way out. Anyways, is that a good enough zoom video for
Yes,
you guys?
yeah, no, well, that's even better.
We can hold this shot for a second, and then we'll get the second going. Let's see, or 8? Or do you want like a 4? Okay,
It's probably going to break up into little pieces, so how about we go for 4 or 3, either of those.
Alright, 3. Let's get the arm going. I imagine it's going to be thin and stuck on the rock.
Yes, it'll just have to break up. And there's a grey sponge just above.
Yeah.
So there's 2 sponges.
Where's the lid cam? Oh, you have to return it on because the second part of it. Turn the camera on. Alright, so we're gonna start with this green sponge right here.
Yes, please. And then
That's off.
and then we've got the grey.
Same drawer or different drawer?
Same.
Same drawer, right. So gray and green.
Should be able to tell them apart.
You gotta get the picture. Let me know when you want section. Okay, I'm sucking. You there. You there. I'm sucking. I'm sucking.
It's pretty hard. You just want to break a piece of it?
Yeah, thanks, Ben.
It's a little more to the right, yeah.
No, I mean break it, not suck it.
If you could.
You get a good sit picture.
It looks like it's just a very thin film. Oh, yeah.
There you go.
Perfect. That's great.
We can get another piece and then I'll do it.
Oh, there's one.
Let's zoom in.
Oh, you got a piece. Yeah. Okay. We can go to the grey one above. That looks like it's.
Alright, you get section. We're still on 3. Okay, break that piece off.
Oh.
The big piece.
Oh, maybe that.
Oh, you
I
can.
thought it was a sponge. It's just grey rock.
Okay, turn it off.
Okay, section's off.
Okay.
Turn it off.
Oh, sorry, crinoid.
Alright, you're parked. I got the starboard ramp extended against the wall.
You gonna look around some more?
Yeah, since we're here.
There's another sponge on the right.
Can you reach the creamy coloured one there? Yeah.
Like I think it was a sponge.
It
The
was?
gray one was a sponge.
The
That
gray?
was the gray one was a sponge.
Okay, grab.
There you go, that's a sponge.
Nice. Yeah.
There you go.
Very good.
Thank you.
There you go. Okay, you got that one. Oh, I should have.
Happy with the 3?
Yeah, yeah.
Any more? Okay.
No.
We're happy with that. Thank you, Ben.
Okay.
That went into 3 or 4? 3.
Yes.
Oh, hi, Doug.
That's where it had it aligned.
Okay. We're gonna use it a lot, so it's so fun.
What's that?
I'm just gonna hold on to it. We'll use it a lot.
Okay.
you. Those are kind of.
I don't, let's keep going.
You're gonna be using it a lot, so it's on the way. I think everything's up to date. All five is over.
Had a lot of visibility problems, so trouble getting transects done.
Yeah.
So,
But otherwise, it's been lots of interesting things.
enjoy. So I see something out there.
Thanks, Ben.
Greg. You want to see stuff?
Oh, yes.
There was one on the left,
Yeah.
on the right. Hello.
I'm joined by. Oh, here we go, Doug.
What is this we're trying to get in?
Little sea stars. Yeah. Had a switch of pilots, so Doug and Scott are now guiding us after great collections by Ben and Levi.
Oh, yeah. I got you. This is gonna be a suction, yeah?
Yeah,
Okay.
we'll suction it.
Which,
and
which jar would you like?
let's
this
put it
one?
in 7, just to save space for the others later.
Okay.
So we've only got one little thing in 7. So.
Okay. Okay. Line up with 7. Trying to get you a good picture here. Okay. All right, we're gonna get that guy? I believe so. Right. Just the star.
Yes, please, if just the star.
Okay. Section.
Oh.
And that look right. Didn't get the star yet.
Great. Thanks, Scott.
Yeah.
You could try the little crab, the squat lobster. Please.
Give you that one, a surprise. Suction's off until you get close. Let me know when you're ready. Yep. Joints here. Okay. Coming in. Alright. Oh, there's the star on the rock, dang it.
Oh.
Didn't know it was on the rock.
Yep.
Oh, you got the crab, and have to see if we can fish that sea star off there too.
Wait,
Got it.
yeah.
Nice.
Very good.
Nice one.
Greg, did you see it? Under,
Oh, gaggle.
under
Yeah.
the crinoids, there is a little
I
octopus.
see the star.
star, yeah.
I saw the squat lobster come in earlier.
Okay, cool.
You just want to look at it? Should we look at it?
Under the crinoid there, there is, I think I saw a little, very little octopus.
What are we looking at?
Hey, wait a minute.
Yeah.
Is that the,
Look it.
is that the squatting then? Is there another squatting?
Oh,
That
one.
one, yes, that one. The
Oh.
octopus? Yeah, look at it. It's so small. No, in the other. Yeah,
Oh,
on the
the
right.
squat lobster died.
Uh-oh.
Didn't get the squat.
There,
Doesn't look like
on
it.
the right.
Sorry, we're getting confused here. Yeah, let's take that because it's.
Okay.
We can still get DNA from that.
Okay. Oh, I saw him going into that.
Yeah, I thought it weird as well. Weird.
Doesn't want to come off there.
Wow.
Oh. You know it?
Oh, you got it peeled off there now.
Hmm.
It's weird.
Yeah. There it is.
So there's a little octopus
Sorry
over
about that. Yeah, it's just to the right. I think it's under here. Right there.
Yes, little one. Oh, poor. That is very small.
We'll give it a chance to grow up.
Mhm.
All right.
Yes, of
Let's keep
course.
going.
He's gonna hold it there. He's gone.
Yeah, we.
He's gonna bring it back a little bit.
Yeah, I'll bring it. Okay.
Happy there?
Well, we could try a transect one more time.
Okay.
This doesn't look too bad right now. So we'll want to go 100 meters.
Okay.
All right,
Just
let's
measure out 100 meters from each guy.
let's start another transect, please.
Following the line here.
Yeah, for the most part, yeah.
Okay. So that's basically six. Okay.
More than scraps is making a mess. Squid. Saw a big one earlier. It's pretty amazing.
Good afternoon on the bridge. That's an updated waypoint for you.
Good afternoon, update, thank you.
It's a different fish.
It's the first one of those
Yeah,
we've seen.
I haven't seen one like that. That's pretty cool.
Cool sponge there with the 3 siphons.
Oh, yeah.
So we're going to do a 100-meter transect here. If we can. We've had to abort the last 2 because of poor visibility or seeing something spectacular like a hook arm squid. We started the dive today at 700 meters at the bottom of the axis of this canyon and took some push course in the mud, and now we've come up about 300 meters over the last 6 hours. And now we're at 400. We've still got a few more hours to go, so we could get up as maybe as shallow as 200 meters. It'll be interesting, especially when we hit the oxygen minimum zone. You can see wherever there's a rock without sediment on it, there's a lot of life, and so it's pretty intense competition for spots. Starting to see more of those fish. Lots of acester clams there, with the white shells with the orange fringe. They're a kind of file clam. A lot of their relatives can swim, and they're closer to scallops as well. Acester are well known from canyons. They're well known for tolerating low oxygen levels as well. Deeper, we're seeing a lot of brachiopods, and only a few acester, but now the brachiopods seem to have dropped out.
What depth are you expecting the oxygen minimums of?
Probably 250.
250.
Yeah. It is certainly dropping now.
Close to it now.
Okay.
Actually, yeah, right now.
All right,
Okay,
that's 100
great.
meters.
Call that the end of the transect. So, how many have we actually finished? Is it 4?
This was the 5th, but the previous one was aborted, so cancelled. So
And the one before was short.
yeah, and it was the last two were from waypoint 4 to waypoint 5. So I don't know if you wanna do another one.
People will leave another one for later.
Mm-hmm.
Especially when we get to the oxygen minimum zone, which we may not do. We'll see. It's pretty steep. We're already at 370 meters. Looks like a skate. Could we stop at some point at one of these clusters? Doug, doesn't
Sure.
have to be this one,
Okay.
but get a nice image of it. Thanks.
All right. You want more than like 2 though, right?
Yeah, cluster.
Okay.
That's what sort of its iconic look of them. We did sample some down, but we couldn't get a good view, so it'd be nice to get something like this. Oh, there's a
How
nice
about
one.
that?
Yeah, that'd be perfect. You can see the valves of the clam are 6 clams there, are wide open. So if they don't get disturbed by us, we should be able to see their gills, their little foot. Yeah, you're able to see right inside the clam there. Beautiful. The shell's very delicate and ringed with those cirri. You can see the big adductor muscle there holding the 2 valves, and that's what people eat for when they eat scallops, is that muscle.
Greg, are we collecting?
Oh.
No, we
okay.
won't.
There is a
Sorry.
Jura. Here.
Sorry?
It's not the Jura, this one, the small one?
Oh, just below? Yeah, little baby. That's a baby spoonworm there, and another one next to it. The body of the worm is down in the hole, and very difficult to collect. And up over here is looks like a fireworm. That's all I zoom. Oh, the macrurid nearly got it.
Greg, do you think that this little graph
Yes, they have the same banding on the legs. Yeah.
And maybe this, because before, during the transit, we saw some individuals that were bigger size and now smaller one. So probably, I don't know, if it's the reproduction season for them. Look at, there is another one.
I'll try and get us a little bit closer.
Oh,
There's a couple of polychaetes in this particular frame if you're a polychaete fan. So there's a feather duster worm there, there's those white tentacles coming out from a spaghetti worm. And then we've had a few of these, they're called unicids, coming out of their burrows. And then over on the left we have the spoon worms. 2 spoon tongue-like structures, clear, white, banded, white margined tongues coming out. And as Anna suggested, the 2 little spider crabs there are juveniles of the much larger libido clear we were seeing all over the place.
What's that green?
I don't know what the green one is.
oh,
It's
it's
moving, it's
moving.
going back in.
Oh,
Oh.
it's back in.
That was cool.
Yeah. And there's limpets here. It's amazing when we have a look what you see. Okay. We'll leave them alone. Head on. Look at, is that hard to get over there and look at that? I think there's a whole lot of feather worms on the wall there. We'll just have a look at this rock slope. There's a lot of filter feeders on it looks like. It's the Acester clam cluster. A lot of bryozoans there, the white branching structures called lace corals. Oh yeah, and then on the wall itself the jelly clear things are sea cucumbers, they're tunicates, sea squirts.
Pause here for a little bit.
Okay, stopping here.
And there are lots of white calcareous tubes, and they're feather duster worms, fan worms. That's a nice one. Some of those feather worms have a plug that will stop up the tube, and we did see one of those earlier, but this one, the red pinky one there, doesn't have any plug at all. So those kind of features are used in the taxonomy for the group. And there's a lot of little brachiopods actually still attached here. I thought the brachiopods had run out, but maybe they're still settling here, but not growing up as well. Kind of interesting the way the Icestas form these little clusters. They're probably individual, they're not clones, they're settled there as larvae, but they may well have cues from adults as to where is a good place to settle. It happens a lot with gregarious animals. So here we see a big cluster of spaghetti-like stuff coming out, and that's actually called a spaghetti worm. And it lives in a tube and it puts out those tentacles to gather detritus. And a lot of the fuzzy stuff, apart from the white branching structures, are actually not animals. They're little things called foraminifera. They're arborescent foraminifera that are covered in detritus. There's a nice view of an Acesta there with the pale orange adductor muscle visible. Okay, that was great. Thanks, Doug. Yeah. It'd be hard to sample those. So the tubes would all shatter. If we saw a rock with some on it, that would be good. The oxygen level is about half of what we had at the bottom, 700 meters, and still continues to drop.
All right, Eric, that's us on the move again.
On the move again.
Seven, our last waypoint? No.
No, it looks like we have more. No, 9.
9, okay. What depth is the top of the canyon plateau out at?
I don't know.
Oh.
It looks like it's past 9 though, because I can kind of see the last
Right.
one right in the yellowish orange
So,
area.
so that's 9?
I believe so.
Okay,
Yeah,
I guess we'll.
'cause, yeah, we're in between, just about at 7. So yeah, I would say that that's 9.
I guess
Assume we just keep going.
Keep going. Yep.
Run out of dive time.
Yeah, how about we start another transect now as
Okay.
we head towards the oxygen minimum zone.
Okay. Scott, can you just mark 100 meters for us and let me know when the transect's over, please? For sure. Thank you.
Okay, we'll do I guess this is really transect, 5th real transect, but
Okay.
whatever you call it. Starting to see much less animals now. A lot more of the arborescent forams dusting the rocks.
So are we marking the start of a new transect?
Yes. So this is our second dive in the Bio Canyon from the Bio River that is near Concepcion on the Chilean coast. This is about, I'm not sure what, the 10th dive of our canyon seeps and vents expedition on the RV Falco with the ROV Sebastian. And we've been at a couple of different seep sites and a couple of canyon sites. We've come in pretty close to Concepcion the last 2 days owing to wind. And we'll be deciding soon where, depending on the wind forecast, where we'll go tomorrow. But in all likelihood we will dive again tomorrow if you're wanting to watch.
Hopefully somewhere with some nice visibility.
Yeah. But not
Although
too bad
this
now.
Probably is sharpening our ability to spot.
Yes. Yeah.
Well, good visibility, but there is no organ like filter. The filter feeders are less.
Yeah, it's bad for us, but good for them.
Yeah.
The rock face there is covered with those white calcareous tube worms. Oxygen concentration's dropping pretty quickly now. I've been expecting to see a lot of brittle stars. We often see them as an animal that can tolerate low oxygen, and often you get a really amazing band in the oxygen minimum zone or on either side of it. And they haven't really shown up here yet, and may not.
You want that transect to be on the horizontal or the vertical measurement?
Mostly vertical.
Because vertical is coming up close to 100. On the vertical we still have, on the horizontal still have 50 to go.
But we've come up 100 vertically?
We combined.
Yep. The combination, if that gives us 100, we can call it.
Okay,
Yep.
almost there. Yeah.
Okay. Yeah, it's just distance traveled.
Okay.
So sometimes they'll structure them as zigzags. You'll be doing a lot of them in the next leg.
Not
Okay.
me, I'm going
You're
home.
going home too? Okay.
Likewise.
And you too, Scott. Okay. Likewise. Yeah, a lot of us.
That's about 100 right there.
Okay, we'll call it. And let's see if there's anything to collect around here. There's a lot of rocks.
Just got my itinerary for the next trip.
Yeah.
When you get that, you're just like, no, not yet, not yet.
I didn't even get home yet.
I wonder if we could maybe take that piece of rock then, if it would come away.
On the bridge, we're going to stop here and sample some stuff.
Nothing here.
We've got about another week on this leg. We're heading south from here to an island that has methane seepage even onshore, and hopefully some deeper seeps offshore, and that'll be where we wrap up and head into Puerto Montt next Monday, and then we'll do a changeover, and the next crew of people are going to keep going south to explore more canyons and vents and end up in Punta Arenas. I wonder if we could break this off. I don't think this rock's, it's like a mudstone. I think it's pretty, it's breakable, but it's whether it would completely fracture. Yeah, the island we're going to is Isla Mocha. Look it up. You can see people lighting fires in the intertidal as the methane bubbles out.
Well, it's free. A little large.
Too big?
We'll see if we can get
Yeah.
hold of it.
Hopefully the crowd will get the message. Leave.
Might be able to get that, Scott. Looks like
Yeah.
a little bit around there.
Oh well.
You wanted the piece with the worms on it.
Yeah, the white spirals.
You might be able to grab it. It's just right there.
Yeah, there's a piece of it that still has them on it.
There's a couple, which one?
The lower one would be good. Thanks.
This one here?
Yes,
Yeah.
please.
I think you got it there, buddy. Nice. Okay.
Perfect. I think any of the back 3 bio boxes would be fine.
You ready? Now with the porch.
Okay.
I'm just coming out a little slow. Oh, wait. You got the cocked up. You got that box cocked.
Getting out of the way. Okay.
Try coming forward first before you swing across. Alright. Okay. Try that. It wasn't closed all the way either, so.
Okay.
Which one will you be shooting for, do you think? 2 or 3?
If I can reach over to 3, I'd like to.
Okay. Well, you're clear now. I can see you're just about clear. And the next thing you got to contend with is the cylinder as the cylinder comes down.
Yeah,
See how you're moving the box now?
Yeah. Okay.
2 is going to have to be it. 2A maybe.
Redo some things here.
Okay. Oh, that seems awfully close to that camera. Must not have the porch out all the way. I can try going out a little bit more. But
Okay, I'm out of the way.
Yeah. Okay, that's all I got there.
Okay, let's just go 2-way.
Alright.
Alright, let it go.
It's in there.
Thank you, Scott.
2A.
Yeah, cheers.
Ready? Porch.
I'm out of the way.
Alright.
While we're here, Doug, we'll have another look around. Just get an idea of what's on the rocks.
Okay.
Thanks.
No problem.
So the white spirals are those calcareous tube worms I mentioned. Then we've got actually a very cute purple pink scale worm there. The branching structures are bryozoans, and there's otherwise lots of brown feathery structures that are arborescent forams. Lower left is a encrusting bryozoan.
Just trying to get us a little closer. Better view for you.
You can see the sepulets coming out with their feathery fans. In this particular one, they actually have the little plug, the one where the scale worm is. You can see that ball-like structure on a stalk that's used to plug up the tube when the animal pulls in.
The umbilical must have settled out. Could have sworn that delta when we stopped here was around 65.
Oh, yeah.
It's a whelk. We've seen that quite a few places. It's a good view of its foot with its operculum on top. And a beautiful little squat lobster.
That is sponge.
Yeah. I think we got some of that sponge earlier.
Yeah.
Yeah. It's really fluorescent. Looks like a sea star down there. Or an anemone. Oh, it's an anemone. It's like got a flower pattern.
That is cool.
Yeah. Baby squat lobster there. The white thing is a chain of mail shell or a chitin. We collected one of those earlier. They have 8 shell plates. Lots of arborescent forams there branching up off the rocks.
That worm.
Anemone. Yeah, little anemone there. It's molt.
Molt.
Yeah, it's
Okay.
molted its
Okay.
exoskeleton. And that's a pretty good view of a feathered osterworm. That has a muddy tube instead of a calcareous tube. Next to a beautiful squat lobster. It looks very much like what we call tuna crabs in California. We often find them in low oxygen environments as well. Got more of those sepulid polychaetes. Can we go up a little bit, Doug?
Sure.
They were hanging on the.
Is that what you're looking at?
Just these things. So we've got some hydroids there and hanging on them are predatory amphipods called skeleton shrimps. These little guys.
Okay.
The bright red
Yeah.
eyes. Yeah. Okay. That's great. Let's carry on a little shallower and then we might sample when the oxygen gets much lower. It's pretty low now. We're at 41 micromolar. So we've travelled up over 400 metres today in depth. It's been quite a interesting series of transitions.
Yeah, on the bridge, we're on the move again.
On the move again.
And now we're hitting the lowest actual level of oxygen. A lot of the filter feeders, a lot of the density and biomass is much, much less here compared to 100 metres down. We were seeing so much more. Can we go and look at those things? Oh, it's line. Yeah.
Greg, can we close on niskin?
Niskin here?
Yeah, because there is this.
What's that, you want this in here?
Yeah.
Is that what you said? Yes.
We might stop and get a rock when it's. Anna, do you want the niskin here or when we stop and get a rock?
We stop close by?
Sure, we'll stop as soon as we see rocks. Why do you like the set?
Because of the fish.
Fish.
I want to
Okay.
see if there is particles.
I see.
And there
Okay.
is.
Okay, so let's do a niskin near that line, please.
You move the camera a little.
Yeah.
Dead crab.
Yeah. We haven't seen the live one of that actually. All right, everybody, I'm going to hand over to our chief scientist, Oilohio Soto, to continue the journey. We're at the OMZ. We're probably going to sample a rock or two now and try for a squat lobster, but we don't want to land anywhere near this line, so we're going to try and find somewhere better for that. Talk to you tomorrow.
Thanks, Greg. Oh, look it. Actually, nice one. The crab, a light one, was here. Look it, here.
Yeah, there's line on. They might not want to see
Yeah, that is a pity. The big crab is here, but there's a line, a fishing line, so we cannot stop.
Hello.
It's a lot of line. They're falling up, wrapping around. So we're trying to not get caught up in it.
What's your ship?
Apologies if I moved you earlier. You're like right across from the line.
If I continue setting off the general alarm at midnight, then yeah, I'm there.
Fair enough. Fair enough.
No, I just, sorry.
Crabs hanging on the line over there.
Muy buenas tardes. Les habla Eulogio Soto, jefe de esta expedición a bordo del Falcon 2, donde nos encontramos explorando el fondo marino frente a la zona de Concepción. Recordar el barco de investigación perteneciente al Schmidt Ocean Institute de los Estados Unidos, liderado en este caso la presente expedición por el doctor Jeffrey Marlowe de la Universidad de Boston y la doctora Patricia Esquete de la Universidad de Aveiro, y también por un grupo multidisciplinario de investigadores del área geológica, oceanográfica y biológica de instituciones de Estados Unidos, Europa y de Chile. Dentro de las instituciones de Chile destacar la presencia de investigadores de la Universidad de Valparaíso, biólogos marinos y oceanógrafos que están liderando esta expedición, investigadores de la Pontificia Universidad Católica de Valparaíso, de la Universidad Católica del Norte y también de la Universidad de Chile. También hay un investigador de la Fundación Biociencia y colegas extranjeros de la Universidad de Nord en Noruega, de la Universidad de Insubria en Italia, de la Universidad del Estado de Oregon, Lafayette en Estados Unidos, abordando investigaciones que tienen como principal objetivo conocer, explorar, descubrir las comunidades biológicas que habitan el fondo marino en el margen continental de Chile, con énfasis en 3 tipos de ecosistemas, los cuales son cañones submarinos. Hoy día estamos en el cañón del río Biobío, filtraciones de metano y también chimeneas hidrotermales. Caracterizando los sedimentos, caracterizando las rocas, mediante estudios de testigos, de columnas, de varios metros de sedimento, también estudiando el agua, la microbiología del agua, el metabolismo, el DNA ambiental, los nutrientes, la distribución de los diferentes grupos que habitan el fondo marino, los organismos que habitan sobre las rocas, las características de las rocas, también los flujos de calor que emanan desde el fondo del mar, las características de las masas de agua, en fin, un estudio bastante multidisciplinario y completo, en el cual se obtienen muestras, se obtienen datos y muchos de ellos también se procesan y se analizan en distintos laboratorios, tanto nacionales como extranjeros. Hemos estado durante el día de hoy subiendo por una ladera desde los 700 metros de profundidad, la ladera sur, en este caso, de un sector del cañón submarino del Biobío, y ya nos encontramos a 238 metros, con condiciones de mínima oxígeno, con una mayor presencia. Vemos camarones, vemos cangrejos, peces, pulpos, pero también una fauna de menor tamaño, muchos organismos incrustantes, organismos infaunales que habitan enterrados en el sedimento, que se adaptan a estas condiciones de menor contenido de oxígeno. Junto a la expedición también mencionar, se encuentra con nosotros un grupo de la BBC de Londres. Son documentalistas que durante este crucero han estado filmando junto a nosotros diferentes conductas, actividades y la vida marina, con el objetivo de posteriormente realizar un documental de los hitos más importantes y posteriormente poder darlo a conocer. Será muy interesante entonces ver el producto final de esta filmación que han estado realizando los documentalistas de la BBC de Londres que se encuentran en esta expedición y que nos han acompañado desde un comienzo. where you can see this area. Bueno, les contamos que hemos estado observando la composición y la distribución de diferentes organismos de distinto tamaño, pero también hay un grupo a bordo que está interesado en conocer los organismos que viven enterrados en el sedimento. Una forma de estudiarlos puede ser obteniendo testigos a través de columnas verticales de sedimento, pero ahora vamos a utilizar un saco o una bolsa y vamos a arrastrarla por la superficie del sedimento con el objetivo de poder recolectar un volumen importante de sedimento. O sea, esta forma de recolección es bastante efectiva, permite obtener un volumen importante y en este tipo de muestras se obtienen bastantes organismos. Entonces es un mecanismo bastante fácil y permite dar una mirada de los organismos que viven enterrados en el sedimento. Así que ahora con el brazo mecánico los pilotos van a deslizar este saco para obtener un volumen más o menos importante, y posteriormente este sedimento se tamiza, se separan los organismos de la arena, los organismos quedan retenidos en mallas y después los organismos se guardan en alcohol y se identifican en los laboratorios. One more, please. Bueno, el grupo que trabaja justamente observando, identificando este sedimento también colabora con el doctor Matthew Lee del Instituto INMAR de la Universidad de los Lagos en Puerto Montt, quien también nos ha comentado que con este tipo de sedimentos es posible recolectar una gran cantidad de meiofauna. La meiofauna es un grupo biológico menor a los milímetros, a varios milímetros, y generalmente los organismos extraídos desde el sedimento quedan retenidos en tamices de 18 micrones y son observados de manera microscópica. Son organismos que viven adheridos a los sedimentos sobre los granos de arena, y que son muy diversos y muy abundantes. Es todo un mundo desconocido este grupo de la meiofauna, que también coexiste de manera muy cercana con lo que son las bacterias, que es un grupo importante que también se está estudiando. Good. You can get that to flip in there, so that's good.
now a bag fell in. You just gotta line that up and let it go. I'll have to see. I have to flip it.
Okay.
Yeah. It's right on the divider.
Como les comentaba, a bordo existe un grupo interdisciplinario de investigadores y estudiantes. Y una de las líneas principales de investigación de este crucero tiene que ver con el estudio de la microbiología marina, principalmente coordinado y liderado por el Dr. Jeffrey Marlowe de la Universidad de Boston. Para contar más detalles acerca del trabajo, nos va a hablar un poco Peter Schroeder de la Universidad de Boston.
Hola, soy un estudiante de microbiología en el laboratorio del Jeffrey Marlowe de Boston University. Estoy buscando en las interacciones de microbios y minerales en el mar, en volcanes y lugares como esto, en los extremos. Entonces es importante porque hay muchas cosas de microbiología que ayudar muchas cosas en sociedad. Por ejemplo, el Cas9 y Taq polímeros están mucho importante por advance médico, y hay cosas más simples como cerveza, queso, vino. Y esos están en los productos de microbios también. Entonces es importante que estudiar las cosas microbiología. Y estoy muy agradecido para hacerlo y para ser aquí con los científicos del mar, como Fernanda a mi izquierda, y también Eulogio y equipo. Es muy importante, y también ellos están más importante. Estamos buscando por microbios, pero en el cañón no hay muchos diferentes tipos de microbios. Entonces estoy ayudando, lo siento, estoy ayudando con los peces y también, ¿cómo se dice? Shrimp. Camarón, sí, muchas gracias. Y es nuevo por mí, pero estoy aprendiendo. Gracias.
Muchas gracias, Peter, por tu valiosa colaboración y por tus palabras para que, bueno, los hispanoparlantes puedan conocer y saber sobre el trabajo que tú y tu grupo realizan a bordo de esta expedición. También está en la sala de control la bióloga marina de la Universidad del Paraíso, Fernando Rego, quien ahora en este momento nos está apoyando con el SILOC, que es una plataforma para anotar la distribución y presencia de los organismos que vemos desde el ROV Sebastian, pero ella a bordo tiene una misión y nos va a contar de qué se trata.
Buenas tardes, bueno, yo soy Fernando Rego y como bien dijo Eulogio, yo estoy a cargo del muestreo de zooplancton, de estos organismos pequeños que están en la columna de agua. Este muestreo se hace mediante la red Bongo. La red Bongo cuenta con 2 paños de red y esta es lanzada al mar
mientras el buque está en movimiento y hace un arrastre oblicuo. Y tiene unos copos, unos frascos que sirven para retener este zooplancton, que luego en cubierta es adormecido para que tenga una muerte bioética, fijado y guardado para su posterior análisis, que en este caso es en el Laboratorio de Ictiología e Interacciones Biofísicas de la Universidad de Valparaíso, a cargo del doctor Mauricio Landaeta. Acá en el laboratorio el zooplancton se identifica, se separa el ictioplancton, que son estadios tempranos de larvas de peces, las cuales también son identificadas, y luego se hace un análisis de morfometría geométrica para conocer el morfo externo de este ictioplancton.
Perfecto, Fernanda, muchas gracias, y muy interesante también para que sepan que no solamente estamos abocados al conocimiento de los organismos del fondo marino, sino que también nos interesa poder caracterizar la fauna que vive en la columna de agua, en particular las comunidades del zooplancton. Otro de los objetivos importantes en este crucero tiene que ver con el estudio del ADN ambiental. Y para ello nos va a explicar y detallar un poco más la doctora Ana Adamo de la Universidad North. A ver, Ana, si nos puedes contar y explicar de qué se trata esto, por favor.
Sí, claro. Gracias, Eulogio. Bueno, la idea es caracterizar los hábitats marinos de profundidad en este caso, y visto que no es posible recolectar todos los organismos que hay, y también lo que no podemos tampoco recolectar lo que no vemos, porque nuestro transecto es nuestra visión durante el buceo es no tan largo en el mismo spot. Entonces la idea es recolectar agua o esponjas o ambas, para luego analizarlo por la ADN ambiental, o sea, todo intentar detectar esa señal genética de los organismos que viven en el fondo marino de este hábitat. Y hemos estado recolectando hasta ahora agua de mar utilizando la niskin bottle, o sea, esta botella niskin que están pegadas asociadas al subasten. Y también es verdad que hemos intentado recolectar cuando era posible trocitos de esponjas, porque son filtradores de agua, porque es la manera en que se nutren, entonces son buenísimos indicadores o herramienta para la ADN ambiental. Y también a través del agua que hemos recolectado estamos también analizando el microplástico, visto que hemos encontrado también muchas basuras en estos hábitats. Y por último, por ejemplo, lo que hemos visto en este buceo, una fishing line bastante larga en el cañón. Y esto es lo que hacemos.
Muchas gracias, Ana, por contarle a la gente lo que estamos haciendo acá a bordo. Hay mucha interdisciplina, muchos grupos diversos, y es bueno explicarle a la gente lo que cada uno realiza, los objetivos que tiene cada uno a bordo.
Good? Yeah. Okay. All right, stop. It's good.
there's something in 8, doesn't it? He doesn't have anything in 8, but it's interesting. That's it? Yeah, it's not really jar. Okay.
Pushing that up.
You're good, right there.
Yes.
Yeah. That was in 4, yeah.
Oh, yeah.
Yes.
Yeah, it's fine.
But I was just saying, it looks like there's something in 8, but you don't have anything on 8.
No, but in 8 we tried to collect the sea cucumber. We couldn't do it because it was attached to the stone, so there is
Oh,
sediment.
okay. Oh, okay.
But maybe we can, when I talk with Greg, we can probably use in future, but I don't know, that's why now the sea cucumber in bio box 4A.
Okay.
Can you make a zoom?
Sure.
Oh, that is the problem.
Yeah, I guess. Whatever it is, it must be delicious.
Yeah, looks like. Because it's not something.
Sí, sí.
On the move again.
Investigate what that is.
Can you make a piece close up here?
Sure.
Thank you.
Healing process.
Oh, look at amphipods. Okay, can we collect all?
What grade are we? We're gonna stop here for a sample.
Nothing here.
Maybe coral quivers, can we collect the whole mass?
Try.
With them?
Can I start right now? Oh, you're on top of it.
Sorry, what was it?
This, we're gonna have to put in, he wants to try and put in a quiver, but it's,
What?
it's this.
Okay.
With the everything?
Maybe.
We do have slurp 2 open. This is the 5 there.
It's all on the back now.
Hey Eric, we're on the move again.
On the move again.
So Ben, he wants to go east of this depth.
Okay.
So, yeah. I can only imagine it's gonna go like northwest. Yeah, just follow the ridge. That way, yeah. Yeah? Okay. Yes, this way. This way.
Basically just follow this
Yeah.
color.
Yes. No problem.
Yeah.
Perfecto. Mate.
The contour looks like 310 degrees. That helps you. Yeah. Can we just stop the ship here? Is that like a good spot for the ship? Then? No.
We're gonna go 310.
Okay, we'll stop the ship. Bridge RV. I got an updated waypoint. We're gonna head to the northwest now.
evidencia de talla de calabaza.
And just about the two cores. These are tons of swell lobsters, like mini swell lobsters.
Baby squads.
Right, so many big swell lobsters.
There's a spider one.
Look at them all.
Up ahead of it.
Okay. There's so many different crustaceans.
The way they all like jump. Yeah.
See that one?
I've never seen this in swamp. It's a lot, look at them all. Zipping around. Or just swimming around. Good, I got an FD. No. Yeah, all good.
Si se ve otro de los caracolitos, se puede hacer eso.
Great, you got an updated
Que se ven como unos caracoles ahí medio grandes. Pero si es que después se ve, hacerle zoom.
Avísame.
¿Ese es un caracol o no? Sí. Mira, no era.
Monse? Sofia?
Son como trozos de cangrejo, parece como muda, ¿o no? No, pero son como muda. Bueno, sí, que se ve el caracolito.
Estamos al aire, parece. ¿Sí? Vamos, adelante estudio, da la indicación el día. Muy buenas tardes a todas y a todos los presentes. Good afternoon. Now we can start talking Spanish.
Or Spanglish.
Yeah. Maybe I'm mixing. Me presento, soy Montserrat González, oceanógrafa de la Pontificia Universidad Católica de Valparaíso, y les voy a acompañar junto a Lucía Villar en esta patita, comentando un poco lo que ha sido esta inmersión. ¿Dónde nos encontramos, Lucía? Muy buenas tardes.
Hola, Monse. ¿Cómo estás? Mira, ahora estamos... Pucha, de nuevo me faltan los lentes. Estamos a 191 metros de profundidad. Estamos bastante someros. Habíamos empezado... Es que empezamos este dive súper temprano hoy día. De hecho, esta ya es como la última patita. Comenzamos en la base nuevamente del cañón submarino del Biobío. Y ya vamos ascendiendo poco a poco. Ya vamos, nos quedan poco menos de 200 metros. Así que feliz de nuevo estar acá contigo, Monse. Con el team chileno a bordo.
Oye, y el team chileno está representado, yo diría, encabezado por una de las personas que nos ha acompañado, o ha acompañado al Schmidt Ocean en los últimos 4...
¿Es titular?
Sí, ella es titular, tiene, ojalá que contrato por siempre. Oye, Maritza Castro, que yo quiero felicitar hoy día porque ayer estábamos hablando respecto a el día domingo. Hoy domingo significa dos cosas: revisión de habitaciones y helado. Yo me quedé dormida, así que ni me revisaron la habitación, ni comí helado. Yo creo que tristeza.
No, pues el helado es ahora, Monse.
Ah, el helado ahora.
Sí.
Oh. No me voy a
Se le iluminó la cara a Monse.
Pensé que no era almuerzo. Bueno, felicidades para mí. Bueno, no revisaron mi pieza, pero Maritza recibió por primera vez en todas sus estadías, que francamente largas, desde enero de este año. ¿O no? Desde febrero, enero. No, desde enero.
Enero.
Desde enero, imagínense esta oportunidad teníamos la gracia de obtener aquel premio dorado. Al fin, que el ticket que te da el premio por ordenar tu pieza muy bien. La verdad es que hay un instructivo, aplausos. Aplausos. Pero entonces preguntarán, ¿qué significa esto? Significa que la próxima semana no necesariamente tiene que limpiar su pieza, pero la próxima semana ya estamos en misión de partida, así que probablemente sí tenga que limpiar su pieza y probablemente a fondo. Así que bien logrado, pero bueno. Maritza, no sé si quieres saludar a tu público presente aquí, probablemente que aún...
Hola a todos, espero que estén muy bien. Y eso, estoy muy feliz por haber ganado mi premio. Y ojalá poder ocuparlo el domingo por último, limpiar el día lunes.
No se pierde la esperanza.
Pero sí, estoy muy feliz porque además hoy es el día del helado. Así que eso, que disfruten el dive y también nuestra conversación aquí a bordo.
Oye, además de Maritza Castro, tenemos también el agrado de presentar a Yerko Castillo. Yerko Castillo está aquí documentando, un poco ocupado, yo siempre lo molesto un poco, pero Yerko Castillo, estudiante de Biología Marina de la Universidad de Valparaíso.
Buenas tardes. Me llamo Yerko Castillo, soy estudiante de quinto año de la carrera de Biología Marina Trabajo en el laboratorio de eventos de la universidad, el cual está a cargo Eulogio Soto. Eso.
Oye, y
Muchas
don
gracias.
Eulogio Soto, que se encuentra con nosotros esta tarde, líder de la inmersión en estos momentos. Buenas tardes, yo no sé si... Veo concentración en esto, pero quizás nos pueda comentar un poco como qué hemos observado en este día, un pequeño resumen. Tengo unas papitas por ahí.
Hola, buenas tardes. Sí, estamos recolectando sobre la plataforma continental de la zona central frente a Concepción unas pequeñas jaivas y también estamos recolectando algunos camarones. Hemos estado realizando durante todo el día de hoy una inmersión en la ladera sur del cañón submarino del río Biobío y nos hemos encontrado con importantes agregaciones de jaivas, pero también de camarones y de otros cangrejos. Así que ahí les van a estar contando y comentando Montserrat y su pandilla más cositas sabrosas del día de hoy.
Yo tengo una pregunta de inmediato, si alguien me la responde. Acá estamos viendo estos cangrejos, estas jaivas como musculosas, a diferencia de los otros días que solamente habíamos visto estas jaivas como arañas. ¿Por qué acá podemos ver más de estas musculosas? ¿Alguien sabe?
¿Algún experto? Se esfuerzan más.
Son más esforzadas las niñas.
Estamos en la zona del gym.
Ah, ya.
Voy a tirar alguna idea.
Ya, tírate.
Quizás porque estamos más someros. No
Y
estamos
ahí
a tanta
habitan
profundidad.
estas musculosas.
Claro, quizás su distribución es más somera que profundidad.
Que las otras que son profundas.
¿Y qué diferencia podemos hallar entre la gente que vive aquí en la zona somera, que es menos profunda, y la zona profunda, así hasta abisal, Yerko? ¿Alguna diferencia que tengan en alimentación, por ejemplo, en esta especie?
No soy experto en cangrejos, pero en la alimentación diría que es la misma o muy parecida. O sea, comen de todo casi, excepto coral y esas cosas.
Pero tienen más acceso a...
Ah, claro, puede que tengan mucho más acceso a alimento más fácil.
Estando en la zona más
somera,
más superficial.
Igual se ve harto sedimento aquí, mira, que va flotando. Bueno, no sedimento, partículas.
No sé si me jugaría por sedimento, más que por partículas.
¿Más que por bichos? Ah.
No, sí, hay harto organismo y harto sedimento. De hecho, anoche estábamos filtrando con la profe Marce, muy tarde, agua, y la muestra, que era de 5 metros de profundidad, estuvo al menos, me la jugaría por 20 minutos filtrándose. El filtro estaba saturadísimo, tapado en sedimento, pero pese a eso el filtro no estaba oscuro, así que no sabemos qué habrá pasado, pero no logramos filtrar la muestra de 5 metros. Pero sí había mucha carga de material particulado, al menos lo pudimos ver en el dive. Y lo seguimos observando hoy. Oye, yo estuve conversando con Ana Damo, que estuvo en el turno anterior también documentando, y me comenta que hoy hemos subido por la pared del cañón viendo un cambio de hábitat y organismos. Primero, muchos crinoídeos, octocorales, braquiópodos, bivalvos, pepinos de mar.
Yeah, a little.
Oye, cambian el equipo, llega Amanda.
Amanda ahora es la líder.
Amanda es la líder. Amanda, say hi.
White, that one?
Hey there. Yes, my name, please.
Oye, hemos visto algo distinto al día de ayer. Maritza, tú que has estado también observando, ahora más pendiente por caracoles.
Los caracoles.
Was it a coral quiver or a suction? Coral. Yeah. That you did pretty good.
Similar a la otra pared.
No.
Or you just want a video?
A Jorge con nosotros.
Oye, sí, Jorge pasa un poco más adelante, sí, hay espacio, no seas tímido, que no te consuma. Yo invito también a la Maritza a sentarse un poquito más adelante porque tenemos espacio. Hoy hay espacio para todos quienes quieran venir a...
Me arrancaron las bolsas.
Oye, con tanta emoción del premio ya entró en desesperación. Oye, pero hola Jorge, ¿cómo estás? ¿Cómo has estado? ¿Cómo van tus muestras?
Hola Monse, hola a todos y todas las quienes estén escuchando. Yo soy Jorge Sánchez, bioquímico de la Universidad de Chile, que actualmente trabaja en Fundación Biociencia. Respecto a mis muestras, van excelente.
No se han muerto.
No se han muerto, exacto.
Espectacular.
Está creciendo lo que no se puede ver en pantalla porque son muy pequeños. Pero al menos el otro día estuve mirando las inoculaciones que estuve haciendo el otro día y se ve vida. Se ve que hay algo creciendo en esos medios de cultivo. Así que estoy contento.
Se cumple la misión. Oye, pero ¿de dónde son tus muestras? Y yo, saludo a Matthew Lee, que se une en la segunda patita, que ahí nos mandó saludos, Jerko.
Un saludo para Matthew.
Un saludo para Mati, que
Saludos
ya
para Mati.
pronto lo vamos a ver, y probablemente ustedes también lo puedan escuchar, o quizás no, en las próximas inmersiones, en la segunda patita que inicia ahora muy próximo, como el 6, yo creo.
Sí, el 6 de noviembre.
De noviembre, así que ahí
Desde Puerto
todos
Montt.
invitados a continuar acompañando esta cruzada del amor a los cañones. Pero volviendo un poco, Jorge, cuéntanos un poco de qué es lo que estás haciendo, de dónde sacas tus muestras, qué es lo que estás buscando y por qué están vivos.
Yo obtengo mi muestra desde los push core, que es lo que se alcanza a ver ahí en pantalla muy abajito donde aparece el número 8, que es la parte superior de un push core, que básicamente es un cilindro de plástico que se entierra en el fondo marino y así se puede obtener un testigo del fondo marino, del sedimento que está presente acá. Entonces yo trabajo con este sedimento a distintas profundidades, tanto de la parte superior, la parte central y la parte del fondo. Y de este sedimento yo realizo incubaciones, hago cultivos microbianos para poder aislar distintos microorganismos que estén viviendo en este sedimento. Microorganismos que estén adaptados a distintas condiciones de oxígeno en particular, o sea, aeróbicos, microaeróbicos, que son de condiciones bajas de oxígeno, y anaeróbicos. Entonces estos son los inóculos que he estado haciendo, que es básicamente tomar una pequeña botella que tiene un medio de cultivo, que tiene características similares a este ambiente, colocar dentro de esta botella un poquito del sedimento y posteriormente incubarlo a una temperatura similar de la cual la muestra proviene.
Oye, ¿qué implica incubar?
Incubar implica dejar la botellita a una determinada temperatura por un tiempo determinado.
Como bajo condiciones controladas.
Exacto. Por ejemplo, si yo tomase un poco del sedimento que se está obteniendo desde esta inmersión, tendré que incubar las muestras a una temperatura similar a los 8 grados Celsius, que es una temperatura bastante similar a la que podemos observar ahora en pantalla, que tiene este sitio. Y además en unas condiciones probablemente de bajo oxígeno, porque como se puede observar ahí, la saturación del O2 es de solo un 10%. Entonces ahí podría utilizar un medio de cultivo que sea, por ejemplo, microaeróbico, que tenga de por sí ya una baja concentración de oxígeno para estos microorganismos que estén adaptados a estas condiciones. Porque también recordar de que si bien esa es la concentración de oxígeno que está en el agua, si no me equivoco, como que está donde se encuentra el rock, el gradiente de oxígeno va disminuyendo a medida que, digamos, vamos haciendo un estudio del sedimento en la profundidad. Entonces el sedimento que está en contacto con el agua puede tener una concentración similar al oxígeno que estamos viendo en pantalla, pero mientras más bajamos en el sedimento, este gradiente va disminuyendo. Entonces hasta un punto donde vamos a encontrar cero concentración de oxígeno.
Claro, con respecto a gradiente es un cambio brusco, ¿cierto?
Sí, un cambio bastante brusco.
Sí, un cambio bastante brusco producido por el consumo, ¿cierto? Porque
sí.
hay muy poco. Yo estoy tratando de calcular cuánto hay en este momento. Porque ahí está en micromol.
Micromol, claro.
Y en ml a ppm me puedo estar pasando de un número grosero, pero 0,65 ml por litro. O sea que estamos en una zona de mínima.
Mínima, exacto.
Increíble, porque igual estamos súper someros. Yo me esperaría ver algo así en el norte de Chile. Fácilmente. Y damos la bienvenida a la profesora Praxe. Si quieres saludar,
hay un
también.
asiento desocupado.
Pero por supuesto, adelante.
¿Qué? ¿Qué cosa? No puede ponerlo.
Muy buenas tardes. Cuéntenos qué hace. ¿Quién es? ¿Qué hace aquí?
Soy yo.
¿Y qué le parece esta mínima de oxígeno que estamos observando en este momento?
Mira, en realidad no me parece tan descabellado. Esto es lo que esperamos encontrar en esta zona de Concepción, que la zona de mínimo oxígeno toca el borde del talud superior y la plataforma en algunos periodos del año. Y bueno, justo ahora estamos en primavera-verano, que es el momento que esto ocurre. A diferencia en la zona norte, esta es una estructura de media agua, no toca el fondo porque la plataforma es muy angosta, entonces no toca los fondos, y eso lo hace particularmente especial aquí porque eso también cambia la fauna que habita el vento.
Uy, hay que recordar que la profesora estuvo aquí acompañando en las primeras horas de inmersión. Algo que note diferente de esas 8 de la mañana siendo ahora las... ¿Qué hora es?
4:30.
A 4:32 de la tarde. ¿Algo que le llame la atención?
Bueno, claro, es diferencia. Es un sedimento mucho más oxidado. Veíamos en la parte más profunda del cañón que las corrientes eran bastante fuertes, había un montón de partículas. Bueno, ahora igual también se ven. Probablemente sea el periodo del año.
¿Qué tan profundo está en la mañana?
700 metros. No estaba en el eje del cañón, pero sí estaba en uno de los flancos, relativamente profundo.
Y hoy estamos en la pared sur.
Sí.
¿Qué quieren atrapar ahora?
O sea,
Estábamos colectando unos caracolitos para el doctor Javier Sellanes.
Bueno, comentarles también que el cañón del Biobío se debe a su nombre porque está en la cabecera, se encuentra con el río Biobío, que es el segundo río más importante de Chile, según el profesor Castillo. Él me sopló ese dato. Dijo que el primero era el río Báquer en el sur, que tenía el caudal más fuerte, y el segundo era el Biobío. Y por eso tenía este tremendo cañón que llega incluso, corta toda la plataforma continental y llega incluso hasta las fosas, hasta más de 4.000 metros de profundidad. Así que es sumamente importante estudiar este cañón. Creo que esta pasada que hicimos por refugiarnos de la tormenta que vivimos antenoche ha servido mucho porque mapearon con alta resolución toda esta área, así que, y ahora aparte de mapearla la estamos observando.
Sí, algo impresionante igual ver las imágenes de Batimeté. Hay que decir que el equipo de geología
Sí.
sigue sin dormir. Yo no entiendo.
Son las máquinas.
No sé cómo lo hacen.
No sé cómo lo hacen. Quiero de eso que ellos consumen. Sí, hoy me costó levantarme. Pero
No
ellos no
tanto.
duermen. Lo bueno es que no me perdí el helado porque aún no ocurría.
Pero cuéntanos acerca de la... ¿Tuvimos una emergencia anoche?
Oye, pasaron muchas cosas anoche. Mientras esta transmisión se terminaba y el equipo aquí revisaba las muestras de sedimentos, se llevaban los organismos y nosotros con la profe Marce estábamos ahí esperando, bueno, y el profesor Manuel Castillo esperando el lance del CTD.